Exchange, Albert Einstein. Good morning everyone. I am Abhinandan from the Department of English, and it is with great enthusiasm I welcome you all to Samagatha 2025, the Intercollegiate Science Fest and National Level Conference on Artificial Intelligence, Technologies and Innovations for the Sustainable Future. We are gathered here today at NLAC to embark on an exciting journey through the transformative world of AI. As we explore its potential for sustainability, let us embrace the innovation, collaboration, and intellectual exchange that will shape the future. Let us now proceed with the inaugural session to invoke divine blessings for this auspicious occasion.
Let us begin with lighting of the lamp and invocation in a unique way. Thank you, dignitaries. To extend a warm welcome to all our esteemed guests and all the attendees, may I invite Ms. Aruna, IQAC Coordinator, MLAC, to deliver the welcome address.
It's a pleasure to welcome you all for the National Conference, Artificial Intelligence, Technologies and Innovations for Sustainable Future, organized by Science Association Samagatha. This conference is particularly important because it is aligned with Sustainable Development Goals of United Nations, adopted in the year 2015 with a call for immediate action by all countries with global partnership to end poverty, hunger, improve health and education, reduce inequality and economic growth, keeping in mind climate change and preservation of natural resources by 2030. I'm delighted that Science Association Samagatha has organized this national conference, which has provided a right platform for students, researchers, academicians, and industry experts to converge and deliberate on AI-driven innovations in many sectors. We are honored to have with us today Dr. Vijay UT as chief guest, whose presence adds great value to this event. Dr. Vijay is Council for Science and Technology, Indian Institute of Science, Bangalore, and he has served in various capacities since 1986. Dr. UT Vijay is currently serving as Executive Secretary of KCST, and he holds a bachelor's degree in civil engineering in Bangalore University, a master's degree in civil engineering from Kuwait University, doctorate from Bangalore University. His areas of interest include water-related technologies such as rainwater harvesting and traditional water harvesting systems, watershed development, application of geospatial technologies for natural resources management, application of 3D laser scanning technologies for archaeology and heritage conservation, specifically digital heritage are some of his areas of forte. Dr. Vijay has been involved in several key projects and programs at KCST, including, as I said, rainwater harvesting, restoration and rejuvenation of Karnataka's Kalyanis, Pushkarinis, Kunte, and Katte, preparation of action plans for watershed development, monitoring and evaluation of watershed development activities using geospatial technologies. One of his hallmark program is Suvarna Bhumi Yojana and dissemination of nanotechnology based water purification for arsenic affected villages. He has contributed significantly to national and international publications through his innovations. He has presented several papers and delivered talks in various national and international workshops and training programs. He is a member of several expert technical committees of various academic, research and development organizations and government bodies. So please join me in welcoming Dr. Vijay. I would like to mention that he has been instrumental in setting up IPR cell in our college, and he has been very magnanimous enough to sanction a lot of projects, student projects from KCST So thank you, sir, so much for your support. So it's a great honor to welcome Dr. KBR Verma, Trustee MLAC and Guest of Honor in his absence. He is unable to join us because of his pre-commitments. Dr. KBR Verma is a distinguished figure in the field of material science. He has earned his PhD in physics from University of Madras in 81, and he undertook postdoctoral research at development of pure and applied chemistry from Strathclyde University, Glasgow, UK. Professor Verma's illustrious career includes a significant tenure at IISc, where he served as professor in Material Research Center. His research interest spans across dielectrics, ferroelectrics, nanolinear optics, and electro-optics. He has focused on functional ceramics, glass nanocrystal composites, and single crystals, contributing to over 200 papers to various international journals. In addition to his research accomplishments, Professor Verma held the position of Vice Chancellor of Sathya Sai University of Higher Education at Prasanthi Nilayam, Puttaparthi. His commitment to advancing education and research is further exemplified by his role as a trustee of Maharani Lakshmi Amani College for Women, where he contributes to the Institute's mission of empowering women through education. Professor Verma's career is marked by a deep commitment to scientific advancement and academic excellence, making him a respected leader and mentor in the scientific community. So we welcome you, sir, in your absence. It's my pleasure to welcome our beloved principal, Dr. Nagalakshmi, ma'am, whose constant support and encouragement is the reason for this program to happen. I welcome you, ma'am. I welcome Vice Principal Dr. Sharmishta in our absence, Chief Administrative Officer Mr. Gangadhar Murthy, Administrative Officer Dr. Anadanesha, Finance Officer Mr. Narayana Subramani, sir, Academic Coordinator Dr. Jolitha, and Science Coordinator Dr. Babita for this program. And it's an honor to welcome Dr. Saudamini, who is on the dais. She is the keynote speaker, and she is from Department of Biochemistry, Biophysics and Bioinformatics from NCBS, Bangalore. It's such a pleasure to meet you again after a decades at having met in IISc. So welcome you, Dr. Saudamini. And we also welcome Professor Yogesh Simhan, Dr. Shiva Teja, Dr. Saujanya, who are going to be taking over the next session as speakers of this particular conference. And very importantly, a hearty welcome to all the participants of this conference and the entire Samagatha team who have contributed in making this conference a reality. Your presence and participations are greatly valued. Thank you so much. Have a great day.
Executive Secretary KSCST as our chief guest. I now request Sir to share his insights with us.
Namaste. Good morning to all of you. Respected dignitaries on the dais, Dr. Saudabini from NCBS, Dr. Nagalakshmi, principal of this college, Dr. Aruna, Dr. Babita, and various faculty members of this great college, and financial and account section officials, and my dear students from other colleges and students from this college, and my dear colleague Mrs. Prajanya, and all dear participants. At the outset, I congratulate and thank the management, principal and faculty members for conducting this 2 days Science Intercollegiate Science Fest and National Conference on very important topic, that is artificial intelligence technologies and innovations for sustainable future. As all of us know that these intercollegiate competitions are very much required to inculcate innovative ideas among the students of various colleges. In this way, Ammani College is conducting almost regular as part of this Kritagna and Samagatha every year by inviting various projects, proposals, post presentations and even essay competition from various colleges across the city, and they are getting good response also from various colleges, especially to showcase their ideas, inventions under science and technology sector. I think this year also just I learned that around 60 competition posters have come from 10 to 15 colleges. It's a good let us congratulate all of them to be part of this 2 days science festival. Like this, we should involve ourselves to think something using science, technology and innovation for the benefit of our society. As we are in the living in the society, we should do something as a student, as a faculty member, as a scientist, as a official. We need to do something contribution to the society where we belongs to. In this way, MLA College has been conducting not only the Samagatha and various such competitions, such festivals, such programs every year. Whenever they approach our organization, Karnataka State Council for Science and Technology, just look at within the IISc campus, just opposite to this college only, we support them. We support them wholeheartedly, not only in terms of financial support, even morally, even technically also we have been supporting this kind of activities, events conducted by this college, because whatever events they conduct with support from all of you, they conduct very meticulously. That is very important thing I have observed that. So in this way, this kind of science festivals, this kind of conferences will emerge as one of the important outcomes in the area of whatever the subject we select as part of the conference. Especially talking about artificial intelligence, AI, we know without AI now nothing is possible. Even today, inauguration of your light itself, Deepa Belago Sameta, we use AI, artificial intelligence. Not only this kind of sector, even whatever in our daily life, whatever the sectors if we take now is AI is, artificial intelligence is playing very important key role in all aspects of our daily life, starting from attendance of students, attendance of our faculty members. AI plays important role, traffic management, even driving cars without drivers also, medicine field also, healthcare systems, navigation, even Google Maps also AI is being used to navigate and to locate the locations. Where when e-commerce, teaching also, voice recognition, face recognition, whatever field if we take now in the era of development, AI is playing very important role, even banking sector also. Each and every sector of our life, our human day-to-day life, AI is playing important role. In this way, the whatever deliberation takes place as part of this national conference, I think you have invited very good speakers from IISc, NCBS and various other organizations. So definitely your participants, that to student community and faculty community who are participating in this today, in this 1 day, 2 days conference, definitely will get good thoughts, and by clearing their doubts and their interests and their inventions by interacting with the research persons, definitely it's going to be a good outcome from this conference, I think, which will help in to sustain the sustainable future. As already Dr. Aruna mentioned, our whole globe is facing lot of challenges, including clean water, hunger, poverty, clean air, energy management, health, various sectors. 17 such sustainable development goals have been identified by our UNO, which has to be addressed by all our nations through inventions and innovations, through R&D, research and development. Then only countries will become self-sustainable, self-developed countries by addressing all these 17 such sustainable development goals. Then coming to our own organization, I am proud to inform you all in this August gathering, our Karnataka State Council for Science and Technology, as an autonomous science and technology organization under Department of Science and Technology, Government of Karnataka, is completing 50 years in this 2025. Thank you. Which was established by the then director of IISc, Professor Satyam Dhawan, the father of space, with support from various professors of IISc, with support from state government. This is one of the first SNT council to be set up in the year 1975. Now, such 35 SNT councils are there, being supported by respective state governments and Government of India, Department of Science and Technology, Government of India, to take science and technology innovations, R&D from lab to land. In this way, in the last 5 decades, 50 years, KSST has been supporting various line departments of the state, especially under the sector of water conservation, by promoting rainwater harvesting and groundwater recharging. Even this college also we have supported technically to install rainwater harvesting in the campus. Energy, promoting various renewable energy sources, wind energy, biomass energy, biogas energy, solar energy, by giving end-to-end solution for renewable energy sources, including biofuel energy. So many programs also have been conducted by in this college also, with support from our bioenergy cell in KCST. And intellectual property rights, in order to safeguard the inventions and innovations of our faculty members, research scholars, researchers in the state from various academic and R&D institutions. KST has been supporting by creating awareness, by handholding, filing of different patents, trademarks, designs, and even copyrights, including GI products of our state. I think now, out of 460 GI products across this country, Karnataka has 48 GI, including your Mysuru Mallige, Hadagali Mallige, Dharwad Peda, then Togari from Gulbarga. Like this, since you have come from different parts of our state, if you find such very important local specific, geographic specific product like GI product, like this GI, please, through your college, inform KSST. Will support, will try to collect various parameters required for registration of these products, in order to promote these products, to give the global recognition to our products which are originated from Karnataka. Like this, we can support this kind of growers, this kind of farmers who are dependent on these products. So like this, similarly, we can support this kind of activities in our state, and KCT has been supporting even in the education sector, student project program, which was started in 1977-78 itself. This is the 40th year of series of student project program, in order to support innovative ideas of our students from science and engineering, including horticulture and agriculture background, to recognize their innovations by giving financial support and technical support from IISc, Government of India and Government of Karnataka, from various other R&D organizations. 18,000 such projects have been supported so far in the 48 years. Even this year also, 1,714 projects have been supported, including 2 from this MLA college. Let us congratulate and thank all the students who have got, out of 3 projects this year, 2 have been sanctioned this year. So congratulations to the respective guide and students of this college. I don't know the display, these 2 projects. So I personally, and on behalf of all of us, let us congratulate those guides and as well as the students from this college. Even next year and also, let us get good proposals from this college. Let us think for the benefit of our society, for the benefit of our people, common people. Let us think and send good proposals from this college. In this way, KCT has been supporting to inculcate science and technology among the students community as well as the faculty community across the state. With this, I once again thank the management and the faculty members for inviting me and conducting this kind of important national level conference. I wish all the best for the grand success of this program. Thank you one and all. Thank you very much.
Words. I now request the dignitaries to release the proceedings of the conference. Thanks to all the dignitaries. Every great institution thrives under visionary leadership. It is now my honor to invite Dr. Nagalakshmi B.N., Principal MLAC, to deliver the presidential address.
First, a remarkable celebration of scientific exploration, technological innovation, and interdisciplinary collaboration. On behalf of MLAC, I extend a warm welcome to our distinguished guests, esteemed speakers, faculty members, and enthusiastic students who have gathered here to explore the dynamic interplay between artificial intelligence and science for a sustainable future. The theme of this year's National Conference, Artificial Intelligence, Technologies and Innovations for a Sustainable Future, a multidisciplinary approach, is both timely and significant. We live in an era where AI is shaping industries, influencing research, and transforming our everyday lives. However, amidst this wave of technological advancement, we must not lose sight of the fundamental pillars of science that make these innovations possible. That is physics, chemistry, mathematics, and biology. Artificial intelligence did not emerge in isolation. It is deeply rooted in the principles of basic sciences. Whether it is the mathematical algorithms that drive machine learning, the law of physics enabling quantum computing, the chemical properties guiding material science innovations, or the biological applications in healthcare and genetics. Every AI is built on a strong scientific foundation. Thus, while we embrace the power of AI, we must continue to strengthen our commitment to basic sciences. It is through the synergy between traditional scientific disciplines and emerging technologies that we can truly drive sustainable and meaningful progress. At MLAC, we take pride in fostering a culture of scientific inquiry, research, and innovation. We firmly believe that a strong foundation in basic sciences is essential in producing future scientists, technologists, and researchers who can bridge the gap between fundamental knowledge and applied innovations. Samagatha 2025 is a testament to this vision, a platform where students, researchers, and academicians can engage in intellectual discussions, share groundbreaking ideas, and explore the limitless possibilities of science and technology. The conference is just not about competition, it is about learning, collaboration, and inspiring the next generation of thinkers and innovators, as rightly mentioned by our chief guest, Dr. U.T. Vijay. So, I extend my sincere gratitude to the Science Association of MLAC, also to our science coordinator, Dr. Babita B., and the entire organizing committee for their tireless dedication in making this event a reality. Special appreciation to our renowned speakers and experts for sharing their invaluable knowledge with us today. To all the young scientists and participants, let curiosity be your guide, let exploration be your passion, and let innovation be your purpose. As we stand at the intersection of AI and scientific discovery, let us remember that technology must serve humanity and science must continue to be the guiding light of our progress. May this event ignite curiosity, collaboration, and a lifelong pursuit of knowledge. I wish all the best for this national conference at MLAC, and thank you all.
Thank you, ma'am, for your motivating words of encouragement. Every successful event is a result of teamwork, dedication, and collective efforts. To extend our gratitude, I invite Dr. Babita B., Science Coordinator, to deliver the vote of thanks.
Thank you, Abhinandan. Good morning, one and all. It's my honor and privilege to deliver the vote of thanks. Samagatha Science Association, MLAC, is witnessing today with a multidisciplinary national level conference on artificial intelligence, technologies and innovations for a sustainable future, involving the multidisciplinary streams of science, humanities, commerce and management streams of MLAC. On behalf of Samagatha Organizing Committee, first and foremost, I would like to extend my heartfelt gratitude to our chief guest, Dr. U.T. Vijay, Executive Secretary, KSCST. And guest of honor, Professor K.B.R. Verma, Trustee MLAC in absentia, for accepting the invitation. I would like to extend my thankfulness for today's keynote speaker, Professor Saudamini Ma'am, NCBS. Technical session speakers, Professor Yogesh Simhan from IISc, Mr. Shivateja Kakileti from Niramal Health Analytics, and Dr. Saujanya, IICT Hyderabad, for taking their time out of their busy schedule and joining us. I would like to thank our Scientific Advisory Committee members encompassing Professor K.B.R. Verma, Trustee MLAC, Professor S.K. Gupta, IIT Delhi, Dr. Saujanya, IICT Hyderabad, Dr. Chitra Ravi, Cybersena Private Limited, Dr. Sankarshan, National Institute of Engineering, Mysore, Dr. Nagendra, MBIT, Bengaluru, and Dr. Shushil Kumar Midha from MLAC. I would like to thank our management, especially to our managing trustee, for their constant support in organizing such programs. I would like to thank our pillar of strength and inspiration, Principal Dr. Nagalakshmi Ma'am, for her support and encouragement and leading the program. I would also like to thank our Vice Principal, Dr. Sharmishta Datta Ma'am, Administrative Officer, Dr. Annadani Sir, Finance Officer, Narayani Subramani Sir, IQAC Coordinator, Professor Aruna Ma'am, Academic Coordinator, Dr. Jolita, for their constant support and guidance in making this program. I would like to acknowledge and thank our sponsors, KSCST, Vasa Scientific, Vanaloka, Ozone Scientific and Ganesh Scientific, whose support has made this event a reality. I would like to extend my gratitude to the organizing team, various committees, stage, registration, proceedings, sponsorship, hospitality, certificate, technical, report writing and media promotion, and especially our student volunteers who have worked tirelessly behind the scene for making today's program a success. A special note of appreciation to organizing secretaries for their dedication and hard work throughout the program. Thanks to the media and the photography team for making a great support. Lastly, thank you for all the participants from various parts of Bengaluru and outside Karnataka for your presence and enthusiasm. I hope this conference will find you an insightful and impactful session. We look forward for a fruitful and engaging session ahead. Thank you.
The inaugural session, we hope this conference sparks insightful discussions, innovative ideas and meaningful collaborations. Wishing you all an inspiring and enlightening experience at Samagatha 2025. Now, I request Dr. Saraswati from Biotechnology Department to introduce our keynote speaker, Professor R. Saudamini.
Who is a distinguished scientist in computational biology and structural bioinformatics. She is a senior professor at the Department of Biochemistry, Biophysics and Bioinformatics at NCBS, Bengaluru, and she also holds the prestigious Mazumdar Shaw Chair in Computational Biology at IBAT, that is the Institute of Bioinformatics and Applied Biotechnology. Professor Saudamini obtained her PhD in molecular biophysics from IISc Bengaluru, and she pursued her postdoctoral research at Birkbeck, University of London and the University of Cambridge. Her groundbreaking research focuses on protein structure, domain superfamilies and plant genomics, contributing significantly to bioinformatics and drug discovery. She is an elected fellow of the Indian Academy of Sciences and also the Indian National Science Academy. Her remarkable contributions have earned her several prestigious honors. It will not be possible to mention all. Just to name a few important ones, she has been awarded the J.C. Bose Fellowship from the Department of Science and Technology, Government of India, and she is also the recipient of the Dr. Raja Ramanna State Award for Scientific Excellence in 2024. With her dedication to advancing science and innovation, Professor Saudamini continues to inspire students and researchers alike. We are privileged to have her with us today and we look forward to her valuable insights. Over to you, ma'am.
First of all, thank the college for inviting me to participate in this very important conference. The AI theme is very pertinent as well for this time. And in particular, I would like to thank Dr. Babita and then Professor Aruna, and also thanks for the very nice introduction. So I would like to introduce to you some areas, especially the biomedical area, where the AI is going to be very useful and requires immediate application. Biomedical area and AI, this combination has been the topic ever since. So I understand that the AI in healthcare assumes primary importance ever since we've been in this AI era. And as it was rightly pointed out earlier, AI is everywhere, and even the lighting was done virtually today. That shows that we are getting there. So what I am going to share today are some of the biomedical areas where there is opportunity for AI. All right. So there are big data available, and there is an immediate need for predictions that are fairly accurate and does not require clinicians and surgeons physical decision making. And also we would require AI to understand genetic mutations. Why are some mutations very deleterious? Why some of them are naive? And we also need AI to predict what sort of drug combinations may have what sort of outcomes, right? And then finally, and most importantly, with the existing healthcare data, such as the patient details, which can be made anonymous so that it can be included for big data analysis, what can we infer? And this sort of analysis is very, very important, because simply when people or clinicians and surgeons deal with their patients, they may not be aware that certain trend is going on. certain communities are prone to certain infections, for instance. Only if we have all the data in one place and perform an unbiased sort of analysis, then we can recognize the trend. But if we use AI, we may be able to also train the AI so that it can go on to a predictive exercise. So I would like to next take forward some of the areas where we can imagine using AI for biomedical data. And first of all, I'm going to explain the large-scale analysis we have done on cardiac patients, publicly available data sets of cardiomyopathies, and how one can focus on certain genes involved in metabolic pathways to better understand the probable connection or correlation between dysfunction of certain metabolic pathways and genes and the onset and prevalence of cardiomyopathies. That's going to be the first story, and then I will very briefly talk about structural modeling of a cardiomyopathy implicated gene, and then I would take you through another story which is on lipid phosphatase, where there are genetic mutations observed in this particular gene, which is a lipid phosphatase, and it has very severe and immediate effect, so much that the children who are born with any of these mutations in the gene either die as soon as they are born, or they require continuous ventilator support. So we have tried to use structural modeling as well as to do some in vitro assays to understand this reason. Alright, so these are going to be the two stories. So you can feel free to stop and ask any questions if you wish during the talk, but after the talk as well. I have the next slide, please. Tightly connected with lipid metabolism and several metabolic pathways. For instance, when the heart goes through a failure, there is a huge difference in the metabolic pathway genes, and some of the pathways that require energy get compensated, and this can then lead to further heart failure. So this seems to be a circularly related event, where the onset of heart failure gives rise to dysfunction of metabolic pathway genes, and mainly the ones that require a lot of energy. Why energy? Because heart, as you know, is a pump, mechanical pump, and it is working all the time, 24/7 for humans and mammals and many species. And as long as the living being is alive, the heart is continuously working. So it is a constantly energy demanding mechanical pump, and the onset of heart failure gives rise to dysfunction of some of the key genes that provide that energy. And this could involve glycolysis pathways, or the ones that use fatty acid, lactic acid, glutamine, and so on. For instance, hypertrophic cardiomyopathy patients see some reduction of the fatty acid metabolism genes, the one that's shown on the right hand side, whereas the other type of cardiomyopathies may be going through some other dysfunction. Okay, so this is why we chose publicly available data sets of different types of cardiomyopathy to understand this phenomena, this interconnection. So the next slide will tell us what are the data sets we had used for the same. And the data sets include, the next slide please. So the next, this shows the kind of data sets publicly available and we had used for the analysis within the first story. And it includes one publicly available data set each of their RNA-seq data, which is purifying the RNA of the patients, and different types of cardiomyopathy. There is hypertrophic cardiomyopathy, there is dilated cardiomyopathy, there is restrictive cardiomyopathy, and there is also arrhythmogenic cardiomyopathy. So this is going to be called as HCM, DCM, RCM and ACM. These are different types of heart diseases that could all lead to heart failure eventually. So what are the differences between them is the question, and can we understand the dysregulation of the transcripts. And in doing this, we have also included some single-cell RNA-seq data that were available for one of the DCM patients, and a couple of microarray results as well. And in doing the, we can relied on RNA-seq. So the next slide will tell us why RNA-seq. The next slide, please. So the RNA-seq is important because it gives a real-time scenario about how many genes get transcribed. Because the DNA contains the genome of a living organism, but not all proteins get transcribed in, the mRNA gets transcribed into proteins, and at a certain event, a certain set of proteins get expressed. And this is very true for the same person, because before food some genes are getting, transcripts are getting expressed, and after food some transcripts get expressed. And if you are under stress, some transcripts get expressed, and if you are say going through an anxiety, some other genes get expressed. And this also differs from person to person, right? For the same situation, one person may appear permanently anxious, but some other person for the same situation might appear super calm. We know that the DNA differences are there, but inherently, depending on the situation, different transcripts get also overexpressed, right? So that's why the RNA-seq is a very good readout, because we can infer for that cardiac patient which has either HCM or DCM or RCM and so on, what sort of genes are getting transcribed. So that is why RNA-seq was chosen, not the DNA-seq. And the next slide please, will tell us what sort of okay, so these are the computational tools that were used to analyze the RNA-seq data, and we had used programs such as Salmon to recognize, to connect it with the human reference genome, because different RNA reads are there, and then we align it with the human reference genome to know what sort of transcripts are getting overexpressed. And then the statistically significant upregulated or downregulated genes are recognized by means of what are called as statistical p-values, and that is derived from a software called PAGE. And then we had used a set of tools called gene level statistics, and this KEGG mapper, for instance, helps us to map the transcripts that have just been associated with different genes by using the previous program onto the pathways. KEGG is a repository of various pathways where genes are operating, and that pertains to the biological function of the genes that are getting transcribed, right? So the KEGG database has been used, and then we again go back to PAGE to attribute significance, statistical significance, and then cell-specific genes can be identified, were identified using the other software called Piano, and then you can use various tools, and we can write our own scripts to then report and understand what are the dysregulated genes. And it goes on like this, but then we also performed a meta-analysis wherein we asked a further question, not only what are the transcripts that are getting upregulated or downregulated in different types of cardiomyopathy, not only what are the pathways they are involved in, are they statistically significant, but also what are the proteins they are interacting with. And that will be important because it helps us to know their influence of the consequence of this dysregulation on the neighboring proteins. Neighboring means not only physically neighbors, but those that are interacting in the pathway. And for this we consulted databases such HPRD, MINT, INTACT and so on. And where available we used kinase data. Why kinase data? Because kinases are important enzymes that perform the catalysis of phosphorylating other proteins, and phosphorylation is literally like an electric switch in the biological cell. Either you have phosphorylation going on or dephosphorylation going on that helps the biological cell to regulate or kickstart some signals and events. right? So the phosphorylation was also used, and using all these calculations, we were able to come up with 4,158 ACM dysregulated genes, and then likewise 5,822 dysregulated genes in the dilated cardiomyopathy, 3,000 and odd for the hypertrophic cardiomyopathy, and then the 1,655 for the restrictive cardiomyopathy. Already you can see that although all of them are cardiac diseases, although all of them lead eventually to heart failure, which I said is a very downhill pathway for the patient, there are differences as to what sort of exact type of cardiomyopathy is going on. So these differences are there, and out of this we selected and filtered only those genes that are known to be involved in the pathways that are themselves connected with metabolic disease, metabolic pathways. Alright, because why metabolism? That pertains to our very first slide, where I had told heart failure gives rise to dysregulation of metabolism, and heart is a mechanical pump that requires energy and so on, right? So the glycolysis and other pathways. So therefore we particularly focused on these pathway genes, and that gave rise to what is called as a differential map or a heat map of getting to know what sort of genes are upregulated, because this color coding, if it is pink in color, it means that particular transcript is highly expressed for that particular cardiomyopathy. And if it is blue in color or green in color, that means that particular transcript is poorly expressed in the transcript. And it helps us to compare and contrast between the 4 types of cardiomyopathies, namely the DCM, ACM, HCM and RCM. These are having different outcomes to the disease heart. Alright, and you can see that a number of genes are actually upregulated in the DCM patients in comparison to the other types. So also in the HCM patients, a complementary set of genes, a variety of other genes are getting upregulated in the HCM patient. How are these HCM and DCM different? Because you can see if it is a HCM patient, their heart is very constricted. So the heart finds it very hard to pump blood. So as if we have gone through a running race. And imagine HCM patient always feel as though they have just gone through a running race. Okay, so they feel palpitations all the time, and they feel as though their heart is going through a huge amount of exertion. And many of you may be marathon runners or just runners. So imagine even though you're good at it and your body and your heart is used to that exercise, surely at the end of your run, you can feel your heart beating, right? So that's how the HCM patients feel all the time. Whereas the DCM patients have an enlarged heart, very ballooned sort of heart. So then they also have problem because their heart is not able to effectively pump blood, because there is no constriction, but the effectiveness is nevertheless different. So why I am bringing this phenotypic differences is because you can imagine how the HCM patient can have completely different set of transcripts compared to the DCM patients. And our objective is to see which one of them are in the metabolic pathways, right? So and therefore you can see that the DCM patients indeed have very particular upregulated genes that are in metabolic pathways, which is not present in the HCM patient. And the HCM patients have their own profile of upregulated genes that are not tuned in for a DCM patient. So that is very clear. So then we mapped it back onto the biological pathways, the metabolic pathways, and we can see that in a DCM patient, metabolic pathways such as thiamine metabolism, galactose metabolism, fatty acid degradation, folate biosynthesis, purine metabolism, riboflavin metabolism, these are all highly upregulated. So in a DCM patient who has a ballooned sort of enlarged heart, what is happening at the cellular level is they suffer from high extents of these proteins that are involved in these pathways in comparison to a normal person or even in comparison to a HCM patient. On the other hand, the HCM patients have indeed less extent of these genes present. The DCM patient has high amount of transcripts involved in the purine metabolism. Purine, as you know, is a nucleotide. Metabolism, the production of the nucleotide. On the other hand, the HCM patient has less amounts of the same genes that belong to the same pathway. In particular, we were attracted by the arachidonic acid metabolism because it falls under the fatty acid and the energy giving cycle. And we had already discussed that for heart failure, if the energy is not sufficient, it gives rise to further heart failure. And therefore, we focused on this particular pathway, which is the arachidonic metabolism pathway, and you can see that it is highly upregulated in the DCM patients in a statistically significant manner. But in the HCM patient, it is statistically down-regulated. One might argue that both of them are patients, so why worry? But we would like to know what sort of changes give rise to what sorts of eventual heart failure, right? Because then the other reason is we could have compared it with a healthy heart, and these sort of samples you don't get easily for a healthy heart, because why a healthy heart containing person will come and give all the way come up all the way up to giving samples, right? So then you have data only about patients, and then you can do a compare and contrast. That's why we have done this. And you can see that arachidonic acid metabolism pathway are very interesting because it's upregulated in DCM patients who have an enlarged heart, and then it is downregulated in the HCM patient who have a constricted heart. Okay, and then we went on to ask further questions such as which of the genes in the arachidonic acid metabolism pathway are going through this dysregulation in particular. And these are all the genes that are shown in the x-axis are the ones that are present in the arachidonic metabolism pathway that gives rise to the synthesis of arachidonic acid in the heart. And these are, in the y-axis you have the different types of heart patients and their samples. And you can again see in the DCM patient, it is the gene called PLAG2G2A is highly upregulated. Here again, red color means upregulation, green color means downregulation, whereas in the HCM patient it is contrast because it is highly downregulated. So you can, I think, appreciate how big data analysis helps us and how AI might help in future to handle these sort of large data sets. And to help you understand, these are only 4 patient samples. But if we had access to more public domain data sets, and there are so many heart patients out there, both in India and as well as generally in the world, so then you can expand it. And this is almost like a pilot study we have done, taking only one type of RNA-seq data from each of the 4 types of patients, heart patients. But there's so much out there, so much promise to be that we can expand it to a larger number of patients, and that means you can no longer do this semi-manually. I'm saying semi-manually because we had used softwares, but imagine doing this for thousands of heart patients. Then handling the data becomes a very big problem, and inferring any detailed signals become very hard. And that's where AI is important, because it can handle thousands of parameters, many parameters, that is the heart phenotype, and then it can look at what genes are there in different cells. And I will later explain to you how single cell data also helped us to understand a bit more. Okay, so that is just to impress upon you that the availability of AI could definitely be helpful. And this is now mapping that different genes in the, which pathway are we talking about? Any student? You want to say which pathway we are looking at now? Arachidonic acid metabolism pathway, yeah. So in that, indeed the PLAG2G2A, which was highly upregulated in the DCM patient and downregulated in the HCM patient, you can try to see at the single cell level in different tissues of the heart, where are they present, all right. And what are the tissues? These are different tissues, monocytes, macrophages, mast cells, and epicardium. And you can see the PLA2G2A gene, for instance, is heavily expressed in the epicardium of the heart. So you can see that how this all brings together, weaves itself nicely to give a meaningful result. That is, if we start to compare the DCM and NHCM patient, we know that some of them are dysregulated between them. And if we try to focus only on those that are involved in metabolic pathways, you bring the number down by from thousands to hundreds, 10,000s to hundreds. That's a huge reduction. And out of them, if we look at trends of whole pathways, whether how much they are affected during HCM or DCM, then you can bring it down to 1 or 2 pathways, and within that you can focus on single genes. And next I'm going to tell you how you can zoom into a gene so much that you can now map the mutations that are responsible for the HCM or DCM and try to understand from structure, which is at the atomic detail, about what is going on, that these mutations give a HCM phenotype to the patient or a DCM phenotype to the patient. And for this, I'm going to simply conclude by saying this is how the analysis was performed before I go to the single gene analysis. Okay, we start from lot of publicly available data sets, and then we look at the focus only on the cardiomyopathy disease, and then look at the differentially expressed genes, the upregulated and downregulated genes, and then we look at the pathways, and then we look at the enriched pathways and focus on the arachidonic acid metabolism, and that helps us to know what are the genes that are interacting with and they also get disturbed. And then you can also map it at the single cell data and get to know whether it is indeed highly expressed or not in the heart, which gives us the anatomical relevance. Okay, that is the large scale data analysis, where I hope I have impressed upon you that with just one patient data for each type, cardiac disease type, you can do this semi-manually, but if you handle thousands of them, definitely high degree of automation and AI could be helpful. So now I was saying that starting from a huge data set, you can pin down to single genes. And one of the other genes that fascinated us, not PL2G2A gene, but the other gene that fascinated us was the gene called CSRP3. And CSRP3 is present in the heart cardiac tissues, and it plays multiple roles. For instance, it could act at specific times to regulate the expression of other genes by physically interacting with a transcription factor called MyoD. And the MyoD, being a transcription factor, it will sit on particular part of the DNA and then promote the expression of genes of interest. So that is almost like a transcription factor interacting protein, fine. But CSRP3 also is a stress responder. So if the heart is subjected to a lot of stress, it's constantly dealing with it. And to give you an idea, say you are in your house, and then if there is a visitor, you make sure that your hall is kept clean, but then you start to rapidly dump all your things in your own bedroom, right? So also the CSRP3 is like a scavenger of stress. And as a result, there is no surprise, I suppose, to imagine that it also builds in and accepts a lot of mutations. That's why we see that in the CSRP3 there are many different mutations that are finally implicated in heart disease. Alright, that being a stress responder, it is more amenable for acquiring mutations and make sure that the other cardiac genes such as the actin, tropomyosin, and then the troponins, troponin T and the C, which are all vitally involved for the heart function, the muscle contraction, are not disturbed. And eventually, however, sadly, the CSRP3 gene also cannot take too many mutations, and that can give rise to heart diseases. And that's the question we asked, and if you map it at the gene level, you notice that there are not one but 2 domains. And domains are nothing but compact regions of proteins, when you look at the structure. And you can see that there is a limb domain 1 and the limb domain 2, and they are connected by a fairly long linker, and this is the structure that shows that. And what we have tried to do is to map those mutations. What are those mutations? Some of the mutations are leading to a HCM phenotype, and some of the mutations are leading to a DCM phenotype. And if you look at the stability, there are softwares that can tell what sort of mutations can affect the stability of a protein to how much extent. Again, going back to your home cleaning scenario, whereas your hall is kept clean, your bedroom should not look too untidy either. So there also you have a selective mechanism you would not like to put all your things on the bed, but you will put it in the cupboard. So also some parts of the protein can take mutations, but some other parts of the protein just cannot tolerate mutations, and it can give rise to a HCM or a DCM phenotype eventually. So it is also going through stress expression. Alright, so these mutations that have been implicated with HCM phenotype, and I don't have to explain what is meant by HCM. We had just discussed this in the big data analysis, right? The contracted heart muscle, which is always going through rapid heartbeat and so on. So the HCM mutations, you can see that they are heavily destabilizing. So if you go and tinker the protein at that site, the protein cannot tolerate it, because proteins are pretty sensitive and they go through a delicate balance of repulsive and attractive forces within the amino acids that are present in the protein. So if you place the mutations at the wrong place, it's going to destabilize the protein so much that it will fall apart. It can open up and it cannot do its function. So these, you can see that indeed the cardiomyopathy mutations are destabilizing in nature. And then we went on to do a saturation mutagenesis on the computer. This is totally virtual. Just like how we had lighted the lamp virtually without ever handling the lamp. This is a manner by which we take the protein, and at every possible site we make a mutation to all other amino acids. And how many amino acids are there? I think you know there are 20 amino acids present in nature. So at every amino acid position we change it to the other 20 types, and then some of the positions it is able to tolerate, meaning these are grey in color, but in some other positions it kind of reports with a dark brown color, which means if you tamper in that region, the protein does not like. Alright, so this again proves us that certain positions, one of them being the mutation itself that I had shown earlier, that is the L44P, which is a disease mutation, it does not tolerate. And at the atomic level, I promised you that I will go at the atomic level and tell you what is going on in a cardiac patient, right? Starting from the RCM, the RNA-seq data, we sieved and then brought it to metabolic pathways, which are hundreds of them. And then we looked at genes like PLA2, G2A and CSRP3. And what exactly is going on at the mutation level? That is what I am going to show you now. Because you see that key interactions are going on in the protein, and that particular region, the L44 region, if it changes to a prolin, which is what unfortunately happens in a cardiac patient, the interactions within the proteins are heavily disturbed. The secondary structures are all falling apart. You can see the wild type has a lot of these yellows, which stands for beta strand or pleated regions of the structure. Whereas in the L44P mutant, if you put it through what is called as a molecular dynamics, which you are virtually trying to move the protein and let it relax to another state, then the key secondary structures are going away. This suggests that the protein, which is wound up and naturally held together, is just literally falling apart. Its key secondary structures are going away. So this, I suppose, helps you to convince yourself that certain amino acid mutations in certain genes can give rise to cardiomyopathy, and whereas some other part of the gene and some other genes and some other pathways may not have a severe effect. Alright, so these again give promise for AI, because we are doing a post facto analysis, a retrospective analysis to provide a structural rationale to say why this mutation is deleterious. But if we have AI, you can feed all the physicochemical parameters, and then the AI will on its own get trained and decide and predict and say what mutations are likely to be disease causing or not, and how this can help in medicine. Because if there is a family that has a heart failure, the grandfather had it, and then the grandmother had it, and the father has it, and then the father has 4 children, we can imagine if we do an early targeted genome sequencing of these 4 children, we can get to know who out of the 4 are likely to be more prone to heart diseases. And we don't have to wait for them to acquire that particular HCM. then do the genome sequencing and then recognize that, yeah, they have gone through a heart disease, right? Because early on we can do a genetic counseling with the family members and make sure that they hold up a lifestyle so that your disease can be either prevented or slowed down or managed to happen later in the life. Alright, so all this can be done with the help of AI. I can promise you this is not fantasy. Already there are publications coming where people are using AI for these sort of predictive exercises. And this is the first part of the story, but already I think that this is the future. And we had Professor Janet Thornton from the UK, who's a key bioinformatics person, and she said the future lies in the AI in healthcare. Of course, the other areas are also extremely attractive and holds promise, but this may be some of the top ones. Okay, so then I will go to the next story, which is a completely different one. So I'm switching gears to a lipid phosphatase, which is called as myotubularin, or MTM1 is the gene name in short. And this particular gene is heavily implicated in diseases, genetic diseases, because you can see that patients who have mutations in this gene are implicated in a variety of genetic diseases. First of all, I'm going to tell you what is the domain architecture. I already introduced to you what is a domain. We saw CSRP3 had 2 limb domains, right? So in this gene that stands for the myotubularin, you have a gram domain, you have a dual specific phosphatase domain, you have a coil region, and then you have a pest region. The pest, for instance, could be for protein-protein interactions. The coil domain can be useful for dimerization of the protein whenever required. And then this is the enzymatic catalytic domain, because that is the phosphatase domain. And the gram domain could recruit or recognize. So I think I should go fast on this particular project. What I'm going to tell is go very fast and tell you that there are as many as 300 mutations that have been harbored by this particular gene that has resulted in this common disease, in which the patients, the babies who acquire mutations in this gene can have very little life. Basically, they die very prematurely, and even if those babies stay on, they require ventilator support because they cannot breathe easily, because their heart muscles are not working, and so on. So what can we do is we selected a set of mutations out of the 300 objectively as to those which have a severe phenotype, those which have this stability values shown by the software, and then something which is usually frequently mutated and functionally important residues. And for these 4 classes of mutations, we took about 5 examples each, and then went ahead and performed 3-dimensional modeling of the key catalytic domain and the gram domain. I have explained to you what these 2 domains do, and then we simply mapped the mutations on the structure, which then gives an immediate understanding about where are the mutations falling with respect to the 2 3-dimensional models of the structure. And they are here. This is a schematic. These are only the selected mutations. These are not all the mutations. And then when you map it on the structure, you quickly realize that they are sitting right in the catalytic domain. Of course, this cannot be a good sign, because catalytic domain is the business end of a protein, and right at the business end, if you have amino acid changes going on, it cannot be very good. And we also performed in vitro assays, that is the phosphatase activity, using a collaboration that we did with our own colleague, Professor Raghu, in NCBS, by which we then asked what are the changes that are going on with the mutations with respect to the one where there is no mutation, which is we call as a wild type versus the mutations. And you can see that the mutations are all having lower free energy, they are less stable. And when we do the molecular dynamics also, the key interacting chemical groups are moving away from the substrate. That is what we see from the structural angle. And when we do the assays, we quickly realize that the lipid phosphatase converts a triphosphate to a diphosphate. So the assay is built around this biochemical feature. And whereas a wild type is able to do a lot of this activity. As much as so much of the product is being generated by the wild type, none of the genetic mutations are able to carry out this activity, suggesting that the key catalytic function is heavily disturbed, and that may be the reason why the patients are having a huge problem. Again, now there are gene therapies going on. There are companies who are trying to help such families by going into the details of the structure and coming with solutions, changing the DNA. And again, AI could be very powerful, because out of 300 mutations, we only selected 20 of them. So we have left behind a lot more, and there is room, because not only MT1 gene, there are so many more genes that cause genetic diseases. Many of them are subcategorized as rare genetic diseases. So the scope is very, very high. So I think with this I will stop and thank the people with whom I have closely worked. Professor Jim Spudich is in Stanford University, and he was the person who helped us to think about the cardiomyopathies in fact, and he has inspired us to take up this analysis. And our collaborator is Dr. Dandapani from INSTEM, which is a neighboring institute, whose lab has a specific expertise on heart problems. And the lipid phosphatase story, I had Raghu, who was collaborating with us. He's also from NCBS, and his student specializes on the lipid phosphatase assay. So with this, I will stop and take any questions that you might have. About two genes in particular. Does anyone remember what are the genes? Anyone? CSRP3, very good, very good. And what is the one that starts with M? Excellent, excellent. So I think you hold a lot of promise, you have a bright future, and I hope these sort of lectures will help you to use AI for different disciplines. And the one that I have touched upon, and that is also a very small fraction, is on the biomedical data.
Opportunity to thank Dr. Saudamini for this wonderful, insightful lecture about how changes at the molecular level, at the genetic level, can help us gain fresh insights into the physiological manifestations of disease. I now request our IQAC coordinator, Ms. Aruna, to kindly present a memento to the Dr. Saudamini.
Yes, Siman sir. Professor Yogesh Siman sir from Department of Computational and Data Science, IISc Bangalore. Now I will give brief introduction about as the research works. Professor sir is a Department of Data Science at the IISc Bangalore, and he is serving as an associate professor. He holds the prestigious Swarna Jayanti Fellowship for the period of 2019-2024, focusing on scalable management analytics of temporal graphs. Sir research interest encompasses distributed data and computing systems as well as Internet of Things. Sir is having a recent publications include research on optimizing federated learning using remote embedded for the graphs, neural networks characterizing the performance of accelerated jets and edge devices for training deep learning models. Professor Sir work has also been featured in the various media outlets, highlighting collaborations such as joint research on blockchain and AI technology between I will get a very good information on data science as well as IoT. Thank you, sir.
All of you over here listening patiently to the various technical talks. What I'll talk about is something maybe slightly different. I'll talk about how machine learning, obviously, it's been extremely well motivated by the previous speaker, Professor Saudamini. I'll talk about how you're able to achieve the performance and scalability required to make machine learning possible through the use of accelerators like GPU and edge computing and so on, right? But yeah, I'm from the Indian Institute of Science right next door, so it is probably the shortest commute I've had in my life going to give a talk. So it's a 2-minute walk. So we are neighbors. And we are very interdisciplinary in nature. So we have sciences, we got engineering, got technology, all of it put together. In that sense, we are a very interdisciplinary university. And the group that I lead, it's called the Dream Lab. We work at the intersection of, I'll talk a bit more about that, which is there in the bottom of this pyramid, and the newer applications that are emerging that can make use of all this computing hardware that's available, or the distributed computing hardware that's available, right? So I'll talk a bit about what's happening in the middle layer. How can you put this hardware to good use for applications such as machine learning, right? And in this, we have a very strong team of PhD students, master's students, interns, project staff who all work on this problem. So it's all due credit to all of them. In fact, the work I'll talk about are done by 3 of my sort of PhD students who happen to be women, Prashanti, Suman, and Rupkata. And they've been doing some really fabulous work in scaling machine learning or applying machine learning to various domains, right? So, and they to me are truly role models for the participation of women in science to advance research and development. All right, so let's talk about why, we're all used to using many of these generative AI models. This is a show of hands. How many of you have used ChatGPT? Right? All of you. If you have not been using ChatGPT, you've been living in a rock for 2 years, right? So, how many of you use ChatGPT or Copilot on a daily basis? Right? More than half the room, right? So, now the question is, who builds all these models? Because behind the scenes, there is a large language model that's running out there, right? Not just one, many of them running behind the scenes, sitting in the cloud, right? So there are 2 parts to it. So someone has to actually train these models, right? Because these models are extremely large in size. These aren't something that you can even train at a university like IISc. You need a lot of computing power, a lot of data for you to be able to train such good models. That's why when China came out with the large language model, it made big news. So what is the name of the model that came out from China last month? DeepSeek, right? So it was a big deal because people thought that only US or Europe having immense amount of computing, the internet giants like Google and Meta and so on could build it. The fact that companies in China could build it was big news, right? Because it takes a lot of computing power, right? So training the models is challenging. At the same time, even using the models for answering queries or to interact with you through a chat is also costly, right? So we'll talk about why it is costly and what are some of the computer science techniques or distributed systems techniques that could be used to make it run faster, okay? All right, so all of us know that machine learning, deep learning is all around us, even before ChatGPT. If you go to social network, if you go to Instagram and sort of, if it automatically tags someone as your friend's name in a picture, that's because there's a machine learning model, a computer vision model running behind the scenes, right? If you see some autocomplete happening in your keyboard, in your smartphone, right? That's because there's a small machine learning model running in your smartphone that's helping you do that, right? Or if you get friend suggestions, right? You go to LinkedIn or some social media, it says, hey, you might want to become friends with ABC. There again, you have graph neural networks or machine learning models running behind the scenes, giving you such suggestions. So ML, deep learning is all around us, has been around us. It's just that it's even more becoming part of our daily lives, thanks to some of the machine LLMs and so on, right? But one of the reasons why such deep learning models is even possible is because of 2 things. There's a lot of data that's available nowadays, and a lot of computing available nowadays to process the data, right? So I remember more than 25 years back when I was in my undergraduate in Chennai, right, doing my BE over there. We had an elective course called neural networks, right? So neural networks have been there for a very long period of time, right? Even back then it had been around for another 15 years or so, right? So neural networks themselves are not new, right? Though they're extremely popular in the last 10 years, right? What's different is that there are these deep neural networks where you have not just a few layers. Back then 3 layers and 4 layers were sort of a big deal, right? 25 years back. But nowadays you have deep networks with like 20, 30, 40 layers and so on that are present. And you can have more layers only if you have a lot of data, right? So because if you have got a lot of layers and very little data, then you can almost memorize your entire training data, right? So even in your classroom, right? What is the right way to learn a subject? Do you want to memorize the textbook or understand the concepts? Understand the concepts, right? So if you have very little training data and a very deep network, it will just memorize the data itself, right? It won't learn the concepts. but you got a lot of data, and the layers are comparatively smaller, then it will understand the key concepts from all that information, because you don't have enough neurons and the network to memorize everything. Since you cannot memorize everything, you sort of learn the highlights, you learn the concepts, right? So that's where deep learning comes into play. You've got a lot of data nowadays, and where does data come from? It comes from all of you. When you go to social media and you upload a picture, you put a post, right? When you use a smartphone app, right? Everything is data being collected, right? By the app, by the social media companies, by the web search companies. All of them are collecting your data, our data, as part of their corpus. But of course, you got more benign data sources. You got Wikipedia, you got just crawling the entire web. So all these are also big sources of data, right? So given access to all this data available on the internet, pretty much for free, right? To all these companies, big data is available for training, right? The second part is big compute, right? And this is where we'll talk about cloud computing and accelerators in the latter part, right? I'm sorry if the pictures are not very clear from further back. It's getting washed out a bit. Can we switch off this light if possible? The light up front. Or is there a switch over here? I think it's more important for students to see the slides than others to see my video, right? No, I myself can barely see. Can you switch off this light? Yeah. The video is for posterity. What's the point of posterity if current students can't learn? Okay, thank you. All right, so hopefully this is a bit clearer, right? So what do we see over here? This is, how many of you have heard of llama, right? Not the animal, the model, right? Llama comes from Facebook, right? So Facebook has put out, or Meta has put out a open source large language model, right? Called llama, right? And that is one of the largest models available in the public domain. DeepSeek is another one also. And they have actually described what steps they take for preparing the data for training the model, right? And all of these data sources are from the public domain, right? So for example, you have this something called Common Crawl, right? Common Crawl is a website that picks up, it's a web service, right? It's a non-profit service. They go and crawl the entire web once a month. And they put that crawled files, terabytes of files, in the internet for anyone to download. So tomorrow or today, if you want to build a search engine, or if you want to build some kind of a large language model, you can go to their website, download this for free, right? And start training your data, right? So that's available. So even Facebook themselves uses Common Crawl data that comes to around, after cleaning and so on, it comes to about 3.3 terabytes of data, 3.3 terabytes of text data, which is a lot of information. You also have a few other sources from GitHub, from Wikipedia, from various books that might be available, PDFs and so on, archive, technical reports, scientific reports and so on. So all these form the corpus, training corpus for Facebook for training their llama model, right? And as part of this, they do a lot of things. They do data cleaning, they do data deduplication, they sort of identify languages, they actually throw away non-English languages, unfortunately, so they only train on English data, and they make sure that sort of things like bad words and so on are filtered out. So they make sure that whatever training data is given to the model, it is more or less somewhat cleaned up data, right? And then they actually form the big training, right? And training itself, we'll talk a little bit more about that. That itself is all this entire pipeline. This is called a data engineering pipeline, right? Before you actually do the machine learning, you have to do the data engineering to clean up the data, prepare it, right, for training, right? So then you build the model, and the models themselves are becoming extremely large in size, right? So we are in the realm of close to a trillion sort of scale model. We are getting there. And most of the models that you see are hundreds of billions of parameters in size. So parameter is almost like, you can think of us as a weight in the edge. We'll have some illustrations of that. But effectively, the more number of parameters, the more you can learn, or more you can sort of, more concepts you can learn and sort of give answers and suggest things and so on. So as you can see, this is probably a year old, right? Right now, this is probably shifted to the right by about 30-40%. But you can see that most of the big companies are trying to build their own model, because it's almost become like a survival game, right? If you don't have among the best models out there, you may no longer exist as an internet company, right? So everyone from sort of Google, Meta, sort of OpenAI, of course, with Microsoft, you have Apple, right? all the big companies are actively competing because they want to make sure that they are not left behind, right? So, if you want to do a web search, which search engine do you use? Google, right? Anyone know of any other search engine? Bing, right? So it's somewhere there in a small corner of the internet, right? At some point in time, when I was in college, right, we had about 10 search engines, right? And soon what happened? People realized that Google was the best, and most people just gravitated over there, and the rest of the search engines died, right? So that's what the internet companies are afraid of right now. If one of these models become really, really good, everyone else is left behind, and they don't have a company anymore, right? So that it's a survival. It's a fight for survival for many of these companies. That's why they're pouring in billions of dollars, right? So if all that money can be spent on education and poverty and sort of other kinds of good things, a lot of the whole world would be in a better place. But unfortunately, it's all being spent on the large language models, right? Because training these models is computationally costly. According to some estimates, the first ChatGPT 3.5 model that OpenAI came up with, it cost them $1 billion to train, right? That's probably more than the entire scientific budget of India in a whole year, right? I need to check, right? But it's a huge amount of money, and all the money is going to the computing horsepower. All the GPUs that they have to buy on the cloud, thousands of GPUs cost money, and that's where the cost goes in, right? All right, but let's talk about how deep learning works, how machine learning works. Machine learning happens very much like how human beings learn, right? So if you think about it, right, when you have a newborn baby, it almost knows nothing. Of course, it's learned something while it's in the womb and growing and so on. But you can think of the neural network in its brain as having random weights, right? It knows nothing. It can't recognize sort of stuff, right? It doesn't know that it's a fan or a light or anything of that sort. It doesn't even know its parents, right? But then you start training it. How do you train it? Every time it sees something, it observes actions and so on. Or you teach it, you say ball, cat, all those things, right? So that's how a neural network training works. So you have a network that you specify. This is the network architecture. You say how many layers that you have. Within each layer, how many neurons that you have, right? And you say what's the connectivity between the different layers, right? That's it. That's pretty much your basic neural network architecture. Again, how do you sort of tune it and so on, and the other kinds of sophistications. But the bare bones basics, this is what you have, right? And from this, what you do is that when you initialize it, you're essentially saying that all the edges have weights, right? These are essentially numbers that you assign to the edges. And what you do is do a series of matrix multiplications. You take the input data could be, in this case, let's say I want to do a classification of an image, right? I take that image and convert it into a matrix, right? I can easily do that because every pixel becomes one cell in my matrix, and that cell will maybe have the color of that particular pixel, right? I can take my image, convert it into a matrix, and I'll take that matrix and I'll start multiplying it with these weights, right? And then apply some kind of a transformation function here, multiply it with the weight, transformation function. I do a series of this, right? It's a series of very simple, high school level arithmetic operations that you perform, right? And at the end, you'll have one final layer, right? You'll have a matrix or a vector in the final layer. And in that, you will say, for example, here I'm trying to do a classification. Is this image a female or a male image, right? In this case, a two-class classifier. So if this weight is higher, I'll say it's female. If this is higher, I'll say it's male, right? That's it. So initially, I have no clue this neural network has no clue how to recognize an image. So it will have random weights. It's like a newborn baby, it knows nothing. Then you send this forward and it will make some prediction, and the prediction is going to be most likely wrong, right? It's a 50-50 chance it will be wrong because it's a two-class classifier. It's like coin toss, right? But once it makes a prediction, then you tell it, if it's correct, you say, shabash, good job, right? So it's actually said that whatever weights you already have, it's actually worked well for this particular image, right? But if it makes a mistake, you say, no, you got it wrong. And then you go to the backward propagation. Backward propagation simply takes the output over here and adjusts these weights, saying that, hey, you have to correct this weight, right? And you have to correct this weight. So you go back and you correct the weights, right? And then you give it another image and another image. So you give thousands of such images, right? And as you go through this image at a time, you keep adjusting the weights such that it learns. That's pretty much all there is. It's just like teaching a child. If it points to a ball, if you ask it to identify a ball, it points to a cat, you say no, right? And then next time you point another image, is it a ball? If it gets it right, you say good. If you don't get it right, you fix it, right? That's what back propagation means. So effectively, you're going to do a series of initialize the model with random values. You guess, doing a forward pass, you make a prediction. The prediction is correct, great. Prediction is wrong. You do back propagation, you adjust the model. And you keep repeating it until you have learned how to classify with good accuracy. Okay? Alright, so again there are different ways in which you do this back propagation. That's one of the sort of tricks of getting this right as to how do you adjust the weights if you get a wrong answer. So that's called, and something called stochastic gradient descent is a popular way of doing that. And just to put this in simple terms, the weight at iteration t plus 1 is going to be the weight that you had in the previous iteration. Let's say you passed an image, and when you pass that particular image, training image, you use the weights WT in the different layers, right? From that, I will actually adjust that with some kind of a loss function over here, right? To get me the new updated weight, right? But typically what I will do is, and you got this alpha, which is like your how fast you want to learn, right? That's a coefficient that you set in terms of a learning rate. But this B term, it's essentially saying that I don't want to pass one image at a time, right? Because passing one image at a time is computationally costly. It's going to be very slow, right? Instead, I'll give you a set of images, right? It's like giving a set of flashcards. Rather than giving one card at a time, you give a pack of flashcards and say, okay, quickly go through all of these, right? So by giving a set of images, set of B images, right? I'll pass this entire set of B images, right? And perform a set of matrix multiplications simultaneously. And what I'll end up with is a set of these loss values, not a single loss value, but a loss value for each of the images, right? And I'll apply all of them, an average of all of them, onto my weights, right? So that's why it's called a mini batch training, right? So how many of you have trained a DNN model, right? Just curious. How many of you have used a DNN model? A lot of you are using it indirectly, but not directly, right? So, okay, so that's what you'll end up doing. So typically, if you have a large set of data, you'll chop it up into smaller mini batches, each having maybe a small set of B number of images. You'll train one mini batch at a time, right? And once you've done the full set of training, it's called you've done one epoch of training, meaning that you've gone through the entire data once. Then what do? This is a bit like when you're sort of studying for your exams, right? Sometimes you might sort of go through the entire syllabus beginning to ending. Sometimes you might actually do it random. You'll randomly pick one chapter, read that, then go to another chapter, right? So that's what you do during training also. You take all these images and you jumble them up. And then again, give it in batches. Right? What this ensures is that you don't end up saying that if I get a series of ball, the next one will be out. You don't want to learn some pattern in the order in which you're showing the images, right? So by jumbling the images, you make sure that the core concepts within the image is learned, not necessarily the order in which you're displaying the image, right? So that's something that you do as well as part of the training, right? So all of this is just a very, very basic primer of how neural network training happens, right? Now, what we'd like to do is operate at scale, right? So, as I said, deep learning is nothing but a series of matrix multiplications, or matrix or matrix vector multiplications. That's pretty much it, right? And have you written code? Has anyone here written code for doing a matrix multiplication? Show of hands. So some of you have, I will see raise hand. So you have a 3 nested loops, right? So you'll actually have to, for each of the cell in your output matrix, let's say it's a square matrix multiplied by a square matrix, you'll actually have to do your first row multiplied by the first column. That will be used to populate the first top left corner of your output matrix, right? So effectively, it's a triple loop that you'll do in, let's say, Python code or C++ or Java code. You'll have 3 nested loops that you do. So the cost of a matrix multiplication is got a time complex of n cube, right? You've got n items, you'll have to perform n cube number of operations to perform your matrix multiplication. So it's computationally costly. The larger your image size is, the more layers that you have, the more number of neurons that you have in layers, right? The more weights that you have, the more parameters that you have, the more costlier the matrix operations are going to be, right? And unfortunately, your CPUs, your normal processors like from Intel or AMD or from ARM and so on, they are not very good at floating point operations, because these are floating point operations means decimal operations, right? So even for all of us, it's easy to multiply whole numbers or decimals. Whole numbers are simpler, right? So even for human beings, whole numbers are simpler, right? Same thing for computers. Whole numbers or integers are easier for them to calculate. If you want to get into decimals, it has separate floating point units, right? So only a small part of the entire CPU can do floating point operations, right? But this is where the second big thing came in. The reason why even deep learning is successful. The first is availability of data. We talked about that. The second is availability of compute. And here, as we saw, your regular CPUs are not good enough. What do we instead use for training deep learning models? What's the processor that's used? It's a GPU. Good, right? So GPUs are used for training these machine learning models, right? And why do we use GPUs? Right? What are GPUs usually used for? GPU stands for graphics processing units, but it's primarily used for gaming, right? Or it's used in sort of all your animations that you see in movies, right? So it looks hyper realistic, right? So if you see a movie like Bahubali, right? So you can trust that the actual superstar is not really lifting a huge rock, right? It's all CGI, right? Computer graphics, right? But it looks extremely realistic. Or if a lion or a tiger comes and pounces on them, you're sure that it's not a real lion or a tiger, but it looks very realistic. And that's all because of these GPUs that are doing a lot of calculations to come up with realistic images, right? Because all of these are called physics-based models. They run these physics-based simulations of the real-world behavior, right? And GPUs are great at it. They are great at doing floating point operations. They're really good at doing math, right? They are like maybe the one kid in class who's really good at math and good at nothing else, right? They're a bit like that, right? So the good news for DNNs, right, or machine learning, is that GPUs were already there. GPUs are used a lot by gamers, by movie studios and so on. So they were really available off the shelf. So machine learning picked up GPUs and said, I'll pass my deep learning operations, floating point operations to GPUs, right? It's because GPUs were available as an accelerator and data was available that DNN really took off in the last 10 years, right? Otherwise, the ideas themselves are very old. You didn't have compute, you didn't have data to make it happen, right? But we are at a point where the sizes of these models are so large, they don't fit in a single GPU, right? Because GPU is very unidimensional. It can only do these math operations, floating point math operations. You can only do, it can't do anything. You can't run a word processor. You can't run a browser. You can't play, you can't do some kind of editing in email or anything using a GPU. It can only do sort of graphics and math operations, right? But it has many, many cores, right? So how many cores does your smartphone have? When you go shopping for a smartphone, do you look for one CPU core? 2, 4, 6? So you'll typically look for, as part of the specs, at least 4 to 8 cores is what you look for in your smartphone processors, right? So because you need to play a lot of games, you need to sort of do a lot of social media, all these apps take up memory and compute and so on. So even in sort of CPUs, normal CPUs, you got many cores. You got 8 or 16 cores is fairly common. 128 cores are also fairly common, right? But with GPUs, you get hundreds of cores, right? Some of the top end GPUs nowadays have 10,000 cores, right? Even in your medium end graphics sort of workstations that you have a couple of thousand cores present in them, right? So these are all good because you can do a lot of compute in parallel, right? So each can sort of give a part of this matrix multiplication to different CUDA cores. In the case of NVIDIA GPUs, you give it to different CUDA cores and they'll be doing the processing in parallel, right? But one thing that GPUs are not good at is that they don't have a lot of memory, right? So typically for your high-end servers that you have, right, that you might find in the cloud and so on, they might have even hundreds of gigabytes of memory, right? Even having 1 terabyte of memory is not uncommon, right? If you got 100 CPU cores, having 1 terabyte of memory is possible, right? But GPUs tend to have less memory, right? They only have about 24 or 32 GB of memory, right? So if I want to perform all these matrix operations, right, all these matrix have to fit in memory, because in a computer, unless something is sitting in memory, my processor cannot access that data and process on top of that, right? So since GPUs have less memory, you are limited by the size of your model that can fit into your GPU memory. So for LLMs, the large LLMs don't fit in the memory of a single GPU, which means you need to have multiple GPUs, right? So that the model fits in memory, right? And that's where things start getting costlier, right? First of all, GPUs are costlier than CPUs. Second of all, you can't make do with a single GPU for these LLMs, right? So there, you need to have multiple GPUs. But now the question comes that I have this neural network model, I have to perform all these matrix operations. How do I do it using multiple GPUs, right? That's where you get into different types of parallelism, right? Parallelism is essentially saying that I want a group of you to solve a problem, right? So what do? Let's say I have a simple task, right? I have 10,000 numbers, whole numbers I've written in a piece of paper, right? I want you to add it up as quickly as possible, right? If you do it individually, it'll take a fair bit of time. Maybe it'll take 10-15 minutes, right? But if you had a team of you, what would you do? One simple thing, you just start the sheet, give one subset of numbers, right? Maybe each page has 1,000 numbers, and I have 10 sheets. Each one takes a sheet, 10 of you, add it up individually, you'll get 10 different numbers. You add up those 10 numbers, you get the final answer. What you've just done is parallel processing, okay? That's what you want to do with these GPUs. Let's say I give you 10 GPUs, right? Then you will actually split this entire mathematical operation into 10 parts and have different GPUs solve different parts and then aggregate the answer together. That's parallel computing for you, right? So now there are different ways in which you can do this. Even in your the sheet that you have, let's say I give you 10,000 numbers and 10 sheets. Let's say each sheet has 5 columns with the numbers, right? I could have taken a single sheet and torn it into 5 strips, right? And for whatever reason, I'll give you the first column of every sheet will go to one student. Second column of every sheet will go to the second student. That's also a fair way of doing it. If you only had 5 students, you could very well do that. There's nothing wrong with that, right? It's a little bit like that even with your parallel computing. You can take all of this. Particularly, you want to do a series of these matrix multiplications. You can either do it, the first layer, I'll give it to the first GPU. The first GPU is responsible for doing these 2 operations. Then the second GPU is responsible for these 2 operations. Third is for GSOP. This is one way of splitting up the work, right? And that's called layer parallelism. Each layer goes to a different GPU, right? But you can also have things called model parallelism, where I'll split it vertically. I'll say this left half of the mathematical operations will go to the first GPU. The middle half will go to a second GPU. The right hand side to a third GPU, right? These are all different ways in which you can split your mathematical operations across multiple GPUs to solve them faster, right? So 2 things. One, you want to solve it faster. More GPUs means more number of cores. The second reason, more GPU means larger memory, which means you can fit a larger language model or DNN in your memory, right? So both are reasons why you want to use multiple GPUs, right? All right, so that's a very quick primer on parallel computing for DNN training, right? Using multiple GPUs, right? So in the last part, right, I'll briefly talk about how some of these, so one part of it is that you can run all these models, train them on the cloud, right? Where you got hundreds of high-end GPUs and so on, right? Which the big companies do, right? But at the other end of the spectrum, I'll talk to you about edge accelerators, which are smaller, more compact GPUs that arguably even some of you can the institutions can buy, right? On which you can do such model training and inferencing, okay? So let's talk about the computing availability more broadly, right? So cloud computing has become very popular, right? So how many of you have used the cloud? Any of you use the cloud? So in fact, all your hands should be up, because as we speak, all of you are using the cloud indirectly, right? Every app that you're using is running on the cloud, right? Of course, some of you might have programmed the cloud. Some of you might have actually gotten access to a virtual machine and run your own code on the cloud also. But cloud is all around us. Just like 4G, 5G, you take it for granted. Your smartphone, you take it for granted. Cloud, you have to take it for granted, right? So if the cloud went away tomorrow, you can have internet, but there's nothing to connect to, because the internet, all your apps effectively go through the internet and connect to these various services running on the cloud, right? All the web pages that you go to are sitting on the cloud, right? Your Google Docs sitting on the cloud, right? If you're doing this on Teams or Google Meet or Zoom, that's running on the cloud. Everything runs on the cloud, right? So cloud is nothing but these large data centers, right? Maybe even bigger than this classroom, right? And you'll have rows and rows of rack servers, right? In fact, if you guys want to do a site visit to IISc, we'll be more than happy to show some of our rack servers that we have in our own data centers at IISc. So you have racks and racks of these, what looks like these bureaus, right? Like your filing cabinets. And within that, you have these blades of servers, right? Each might have about 20 or 40 servers in one rack, right? You might have hundreds or thousands of these racks, right? So what data clouds do is they consolidate computing power, right? It's like a warehouse. You consolidate thousands of servers and you rent it out, right? So a cloud is nothing but a rental business, right? So you go and say, for you to purchase a server, typically at any large institution, it will probably take a month or two to complete your purchasing process, right? So the process itself is very tedious, never 100% utilized, right? At best, it might be 50% utilized, right? Because it's very rare that people are going to be running jobs in the middle of the night, right? Of course, some students will be doing that, but half the time, even in your own labs, right? Your computer labs that you might have over here, you're only using it maybe for 5 to 10 hours a day. The rest of the time, the machines in the lab are lying idle, right? So that's one of the challenges with buying hardware. What cloud allows you to do is rent hardware. And you can rent hardware almost on an hourly basis. If you want to rent 10 servers for 1 minute each, you can do that today, right? And you'll only pay for 10 server minutes. And the cost for that might be as small as a cup of coffee at Cafe Coffee Day. You might pay 200 rupees to rent maybe 10 servers for a few minutes on the cloud. So what that means is that most of the companies, organizations, enterprises have shifted away from buying and maintaining hardware to running things on the cloud, right? So cloud computing is a first-class computing entity in most organizations, right? But you also have some degree of private data centers where you already might have legacy data, or you might have very legacy servers, you might have sensitive data and so on, that might still be sitting in your local data centers, in your own institutions. Those are called private clouds. And top of that, you can also have cloud data centers in different regions. Even within India, you might have dozens of these data centers, or at least 3 or 4 from the major players are present, right? In other countries also, you might have different data centers. And now with more emphasis on data privacy and data regulation and so on. For example, in Europe, there's this GDPR, right, that prevents you from moving data of European residents outside the geographical boundaries of Europe, right? So you can't take a European resident's Facebook data and move it to the US. That's not legally allowed, right? So which means that you might have to use data centers across the world for any large MNC to use the cloud also, right? On top of that, what you're starting to see is this notion of edge computing, which I'll talk about as yet another type of computing infrastructure that you can start using, right? So what you're seeing is a computing continuum, so where you got big data centers sitting in far off regional places where you can remotely access, to more locally accessible edge computing devices, right? So anyone recognize what this what this computer is? This green one over here. So anyone use a Raspberry Pi over here? So Raspberry Pi is a very compact computer. It costs about 5,000 rupees. It's probably the size of your smartphone, maybe slightly thicker, right? But it's a full-fledged Linux machine, right? You can connect an HDMI cable, a USB keyboard, and so on, connect a monitor, and you're done. You're actually got a full-fledged computer. Right? So that's an example of an edge computing system. What you're starting to see is slightly more powerful, similarly small in size, but slightly more powerful edge accelerators. This is an NVIDIA Nano from NVIDIA has a Jetson series of edge devices. This is called a Nano. This is about the same size as a Raspberry Pi. But this has about 100 to 200 GPU cores, CUDA cores present in there. A single GPU card with that, right? And this is a more higher end version. This is an NVIDIA or an AGX that's got about 2,000 CUDA cores. That's enough computing power that a full-fledged GPU workstation, the ones with colorful lights and so on you might have seen, it's as powerful as that, but it's the size of, it's smaller than half the size of this projector, right? So these are the kinds of edge accelerators that you're talking about that's coming online and which have fairly sort of strong specs. Like the Orin AGX has 2000 CUDA cores, it's got 32 to 64 GB of GPU memory. Like I said, 24 GB is sort of a high-end workstation GPU. This has got 32 GB, which is almost approaching a server grade, right? So compact form factor, only about 60 watts of power. 60 watts of power is the same power load as my laptop. So using the power of my laptop, as in power consumption of my laptop, is able to run, and the size of a small external hard disk, it's going to give you the power of a computing power of a GPU workstation, right? So we've been playing around a lot with this for both for deep learning training as well as inferencing. And what you see is that there's an exponential growth, right, in the speed of these machines, right? How many of you have heard of Moore's law over here? So Moore was one of the co-founders of Intel, right? And long back, about 30 years back, he essentially made an observation. It's not a law. It's many things in computer science are called a law. They're not a law. They're just an observation, but all the news media sort of calls it a law because it sounds cooler, right? Moore essentially made an observation. He said that the computing power is going to double every 3 years, right? Effectively, he said the number of transistors double every 3 years. But the loose interpretation is that the computing power will double every 3 years, right? And what that means is that there's an exponential growth, right? If we saw such exponential growth in our real life, it will be great, right? So think about it. In 2 years, if you would double your wealth, right? All of us will be crorepathi's within a year, right? So what you're seeing is that same kind of Moore's law in the context of Jetson devices, right? We had one of the early Jetson devices, TX1, and that had barely tens of cores. If you look at computing sort of flops or trillion floating points, it barely had about 1, right? And now the latest one, Orin, has 100 of these T flops that you can perform within a 6 to 7 year period, right? So this is the exponential growth in the speed with only a linear increase in the power consumption. You've gotten from about 10 watts of power to about 50 watts of power consumption, right? So that's a favorable metric, and more of these devices are coming out there. And you can sort of see these deployed in various smart city projects. For example, we did some work a few years back where you could have sort of air quality monitors that need to be calibrated on the field, right? Here again, you can have an edge device sitting in the field, right? That takes in these data and cleans it out before you go send it back to the cloud for further processing, right? So as part of this, we have looked at a few things. I'll touch upon a few slightly more researchy aspects just to give you a feel for what kind of technical problems are here. There are a bunch of papers that we published. You can read up more about that over there. But typically when you're training your DNN model, so you'll load the data from disk, your training data, the mini batches, you load it from disk into memory. Then before you actually run the training on the GPU, you'll do some pre-processing. Let's say if it's an image, you might do some cropping, you might do some standardization, normalization. Those things are typically done in your CPU, right? And then you actually do a series of these matrix multiplication operations on the GPU, right? So this is how your training pipeline looks like, right? So what we see is for the edge accelerators, some of these things are different from your standard workstations. Right? Your disk might not be your standard hard disk or your SSD. It might be an SD card might be present in some of these edge devices. Your memory again is a low power memory because your power consumption has to be low. It's a low power memory, so it's slightly slower than your server grade machines, but it also has more memory. Like I said, 32 to 64 GB of shared memory between CPU and GPU and so on. So there are a few of these differences in these that make working on this edge devices interesting. But one key aspect that we've also looked at is sustainability, because one of the key challenges with AI nowadays, it's costly, right? Monetarily, that's one part. But it also consumes a lot of energy, right? So, which means that doing a Google search is probably 10 to 100 times more efficient, energy efficient than asking ChatGPT for the same answer, right? So, you're putting in, and we are sort of almost like this, some estimate that says the amount of energy going towards some of these is the size of what the power consumption of a country like Poland, right? So, an entire country's worth of energy is being sucked up for training and inferencing on these AI models, right? So what we are also looking at is, can you trade off performance for energy, right? Are you okay with withstanding a slightly, instead of getting an answer within 1 second, if you get it in 1.5 seconds, but you are able to do it in half the energy cost, right? Are you willing to do that? Would you be willing to do a sustainable AI? That's something that we are exploring. So this is showing what's the time taken for getting, doing some of this, in this case training, versus the energy consumption. As you can see, as the time taken sort of increases, your energy consumption also can decrease, right? So that's something that we are observing, right? So that's something that we want to play around with, or we have been playing around with. We also have other things where you can do ways in which you can predict how long it's going to take to train, right? So let's say you start off training, so you want to know whether it's going to take you 15 minutes or 15 hours or 15 days to train, right? So even if it's 15 hours, it's going to be 15 hours or 16 hours, right? Because you pay for some of these GPUs by the hour, right? On the cloud, right? So whether you pay for 15 hours for 100 GPUs or 16 hours for 100 GPUs might be the difference between 15 lakhs and 16 lakhs of rupees, right? And that you want to sort of budget ahead of time so that you don't break your budget, right? So we are looking at models for predicting how long the training time is going to take, how the energy consumption is going to be, and so on. So you can be more sensitive to that. I'll skip past some of these things. We're also looking at how can you run multiple training and inferencing simultaneously, because at some point in time you might want to keep doing inferencing continuously, but periodically you might want to do retraining. So how can you do both of these in the same hardware, where you can give some time slices for training, some time slices for inferencing, while still meeting maybe latency goals, doing inferencing, individual inferencing quickly so that people are not, who don't get bored. But at the same time, the throughput for your training should also be high. So we're looking at problems like that, right? And also looking at other accelerators. CUDA is one of the accelerators. There are other things like DLAs and TPUs and so on, which we are looking at as well, and tensor cores. And lastly, with this I'll go to the conclusion slide in the interest of time. What you see with some of these edge accelerators is they are large enough to even run some of your medium or small scale LLMs, right? Small LLMs is oxymoronic. Small large language models doesn't make sense. But effectively, you are running these large language models that are not hundreds of GB in footprint, but tens of GB in footprint. So you can call these as small language models. We actually were able to, we did this last month. We were able to run DeepSeek on the edge accelerator, right? And get answers out of that, right? So, in fact, we were only planning to run Llama on that. But a week before we had to publish a paper, DeepSeek came out, and we knew that the first question the reviewers will have, why didn't you try DeepSeek? So we actually spent a week getting DeepSeek running on one of these edge accelerators that you saw. And in this case, it had about 32 billion parameters, right? Again, there are different sizes, and we ran that. And we were able to get, in this case, a latency for execution for this DeepSeek. It's a diamond over here. Which one is the diamond? It should be somewhere over here, right? It took about 60 seconds to get an answer out. Of course, you don't want to wait for 60 seconds. This is for larger batch size. So if you give it 128 prompts, right? If you give it a question with 128 words, it'll take about 60 seconds to give an answer back, right? But you're running the whole thing on a small machine sitting in your desk. You're not going to the cloud. So you can be in a forest with this device connected to a USB charger, and you can happily talk to it, right, without talking to the entire world, right? So these are the opportunities that exist. You don't have to have a huge GPU, you don't have to have cloud and so on. And some device like this costs about, well, it's not cheap, right? It's about $2,000, about $1.5 lakh, but that's still much more affordable than having a huge GPU machine for running these things, right? All right, so there's also things like federated learning. I won't get into it in the interest of time. This is the last thing I'll leave you with. How is this practically useful, right? So this is Indiranagar. This is a traffic junction in Indiranagar, right? So if you've gone around Bangalore, maybe if you're going to go to Marama Circle, right, you'll see all these safety cameras, right? And in Bangalore, you have anywhere from 5,000 to 6,000 safety cameras, right? So where does all this, how is all this data put to use, right? It's not put to use a lot, right? The government has spent a lot of money, right? They do some degree of challans, right? So for some of the key junctions, all the cameras that are present at the traffic signal, they actually do automatic challans, right? So that's being done. But for the most part, this data is just sitting idle, right? So if there is some theft or some burglary or some sort of car gets stolen and so on, then they'll go back and look at that, right? But 99.9% of the data is just lying idle, right? So what we have is a project that's starting off. Can we use these deep learning models that we are talking about to actually analyze all this video that's sitting there, petabytes of data. It's about a month's worth of video takes about petabyte of storage, right? Can we look at this and start getting estimates of the traffic flow? Can we use this for mobility analysis, urban mobility analysis, right? And what you see is a variant of YOLO which we've retrained and trying to estimate, right? What is the traffic flow pattern? So what's the traffic flowing from which direction to which direction at what point in time? What is the type of vehicle and so on? We're trying to get counts and classifications and turning patterns for entire Bangalore, right? Once you do that, you can give a nice dashboard. Let's say Google Maps has a nice traffic flow dashboard. We are trying to build a dashboard like that for the Bangalore traffic police, right? And using that, we also want to do predictions, right? If the traffic is so-and-so right now, can I predict how it's going to be in the next 5 minutes or the next 10 minutes, right? Can I predict if there is a unexpected traffic jam? Because Bangalore always has traffic jam. But if something is unexpected, instead of having 50 vehicles per minute, am I seeing 70 vehicles per minute, right? Can you alert the police, right? So this is a practical use of using these DNN models for something that will benefit our day-to-day lives. Right? Because do you know how in worldwide traffic, do you know what's the rank of Bangalore? We are number 6 in the world, right? And 2 years back we were number 1, right? And all that the traffic police did was they reduced the congestion by 1 minute on average, and we went from number 1 to number 6, right? So that's the kind of, so we are number one in the top 10 for the wrong reasons, right? So we are trying to see if you can use some of these analysis to make things better, right? And here's where we want your help, right? So one thing we are doing, so 5,000 cameras is a lot of data, right? And for you to train these models, you need to have not just data, you need to have annotated data, right? Data that actually has labels present in there, right? And for doing that, what we are launching is a hackathon, which is designed as a game, right? So this is going to be launching in the next week or so. I'll send you some of the flyers. It's going to be running for 1 month in the month of May, right? And we want to sign up 1,000 teams from across India, right? And the job is to actually play a game where you'll go from the outside of Bangalore to the center of Bangalore, actually to be precise, to IISc and your neighbors, right? And try to sort of, as part of it, in the game, you'll be labeling the various images that you saw. So you're essentially saying, hey, this is a car, this is an auto kind of thing. And as you keep labeling it, you'll get points, right? And you sort of, as you go through the different levels, you'll get closer to the campus. And the grand prize is going to be internships at IISc, right? So I'll share details of that. But part of the outcome of this is that we want to build the largest annotated traffic data set for India, right? Because currently the largest data set has about 1,000 images. We want to have 1 lakh images that's there, and it will be put in the public domain, right? So that if anyone in India or around the world wants to build a better traffic prediction model that can be used in the Indian context, we want that to be flourishing, okay? So I'll share details of you. Hopefully all of you can participate. It doesn't take a lot of work, and we made it into a game so you can compete with various teams across India. So we hope it will be a fun exercise and you're contributing to open data sets as well, right? All right, with that, I'm going to sort of conclude. So there's a lot of interest in the area of scalable machine learning, using systems for machine learning, parallel computing for machine learning, cloud for machine learning. So do read up on that, so you can always be users of ML, but also understand how ML works. So I've just given a very basic primer, but there's enough material online, right? So you don't have to wait for to take a course on machine learning, right? Or scalable machine learning. You can just go to Wikipedia, read up on it. You can go to Coursera, take mini courses. You can go to sort of any blog post, right? So and read up on that. There's a lot of resources online. You can try it out yourself on your own laptop or your desktop. You don't even need like an edge accelerator and so on to play around with these things, right? So with that, I'll stop. So thanks a lot for everyone, and I'm happy to take questions if there are any.
Understood how deep learning practically worked, especially for BCA girls? You are asking, ma'am, always theory. This is the practically how traffic control they can did, the count of vehicles also. That is also one of the idea for your major project. I hope anyone will take for you, right? Okay, thank you so much for wonderful informations. So, few minutes. I request Babita madam to give token of love to sir. Dr. Sivateja Kakidleti, sir, to come on dais. Madam Principal Research Scientist, Nirmai Health Analytics, Bangalore. Welcome you, sir, for Samagatha 2025.
Thanks for inviting me. So today I'll be talking about how we can transform healthcare with the use of AI-driven thermal imaging technology. First, I will give a brief introduction to what is medical imaging. And yes, I think previous speaker has briefly covered artificial intelligence and how we can apply. Now, I will move on to how we can take any AI-based healthcare product into clinical implementation, followed by how we have developed an early stage breast cancer detection using artificial intelligence. So, what is medical imaging? You might have heard radiology on these terms, right? It's nothing but some kind of visual representation of body parts in order to take any kind of medical intervention or any kind of clinical based management. So in order to get these medical representation, we use different imaging techniques. One of the most common being X-ray imaging technique, which I think one of you or or family members would have gone for some kind of X-ray imaging. What X-ray images does is that you pass high ionizing radiation, and there is a film which captures this. Whenever there is any dense structures or hard structures, they absorb this X-ray radiation, and if it is a soft tissue, it passes the radiation. So it is very good if you want to understand the density differences. So if you want to analyze whether there is a fracture or not, bone fracture or not, X-ray imaging is a good tool. And if you increase the ionizing radiation, we can even measure the tissue density differences, using which we can detect whether there is a breast cancer or not. CT scan is another type of X-ray imaging modality where we concentrate on slices of the different body parts. Ultrasound, where we use high frequency sound waves. We shoot the sound waves and then we collect the echoes. Depending on the intensity and the time delay that happens, we try to analyze the structural properties of the body part. So it's very good if you want to analyze moving parts, or if you want to study the structures of a cancerous tissue. So it's very good at that. MRI is another imaging modality where we use magnetic waves and magnetic field and radio pulse in order to reconstruct the soft tissue in a better format. The X-ray imaging is very helpful if you want to analyze the hard structures. MRI, it analyzes soft tissue very well. Typically it's used to study brain or any kind of breast soft tissues. We can analyze with MRI. So the three imaging modalities which we discussed are more in terms of morphological imaging. They're not giving any information about what is the activity of the tissue. So if you want to analyze what is the activity, the common imaging modality that we use is PET imaging or SPECT imaging, where we inject radioactive tracers and then we collect the gamma rays that's emitting from the body in order to understand how the tissue is behaving or what is the metabolic activity of the tissue. So what are the challenges with medical imaging, right? The first and foremost is that we have millions of pixels to analyze, and for a radiologist it is very taxing. And at the same time, in countries like India, we only have one radiologist for 100,000 population. And the results significantly vary with the expertise. There are studies which show that if it is a junior radiologist, the accuracy could be 60%, and if it's expert radiologist, accuracy could be 90%. So it can significantly vary depending on the expertise of the radiologist. So that's where artificial intelligence can help in. I think the previous speaker, Yogesh, has clearly articulated what is artificial intelligence. Just to understand the definitions, AI is a field which deals with computer algorithms that can learn on its own. Right? It can perform any kind of simple task to a complex task. So machine learning is a branch of AI within AI, where computer systems are capable to learn without being explicitly programmed. So we use something called as training data set, and we try to understand the relationship. Whereas deep learning is inspired by how human brain is connected. So specifically in healthcare, AI has been instrumental in different areas, starting from disease diagnosis. Now we are seeing the use of AI in automatically diagnosing whether a person is having cancer or not. We are able to triage whether a patient is having any kind of abnormality, or we are even saving radiologists time by prioritizing which patients require immediate diagnostic help. Like, take an example of UK. Typically it takes 1 month to get a report. So during this 1 month, the disease can progress. So if we follow the routine workflow, the person who takes the imaging first gets the report within 1 month. Right? Now we have AI systems which can interfere here, and then they can sort based on the risk. which could save the time to get report for a high risk patient in a quicker time compared to a regular 1 month reporting time. It's been instrumental in treatment monitoring, where with the use of AI, we can predict what type of treatment we can give and what are the chances of success. During COVID-19, we all have seen how AI has been instrumental in speeding up the drug discovery process. Digitizing health records is another area where AI is helping in digitizing the health records, at the same time taking this data and then coming up with some kind of risk assessment. And with the advanced robotics and AI, now we are seeing precision surgery is another area which is booming. Telemedicine again, where artificial intelligence is being helpful in automated prescriptions and helping the radiologists. And ultimately, what we can see is the world is slowly moving from one size fits all model, which means instead of taking a generic model, we want to develop more personalized models. So how we can do that is having more data and then trying to analyze with the help of AI algorithms. So if you are working in artificial intelligence in medical imaging, these are some of the 4 common problem statements which you see. So the first one is classification problem, where you are given an image and you want to classify maybe here whether this lung X-ray image has some kind of tuberculosis or not. So this is a standard classification problem. Then there is other set of problem statements called localization problem. So you know that this is a tuberculosis, but where exactly is the tuberculosis region? And since we are dealing with a medical data where we have time series data also is available, and many times when we are capturing these images, the frames are not aligned. So as you can see on the left hand side is the actual captured image where the patient is moving and the region of interest what we are seeing is also moving, due to which the doctor might not be able to interpret how a particular region is varying over time. But with the help of AI, we can register. The right side what you are seeing is a registered frames. Now with the help of registration, the doctor can analyze how the heat pattern is varying over time. So again, because we are using medical images and because we are dealing with multiple slices of images, the next common problem is, can we reconstruct from these slices the 3D body part, or it could be because of the different sensors we are using, can we denoise the image? So these are the 4 prominent problem statements we see in medical imaging. And there are other exploratory problems like how image synthesis, like with the Gen AI algorithms, how Gen AI can help in medical imaging is one area which research is actively happening. So you can assume that Gen AI can help in training junior radiologists, because many times if you're talking about cancer data set, it is very hard to get different cancer images. Now with the help of Gen AI, we can generate different varieties, using which we can train junior radiologists to train them better. And with the combination of NLP and medical imaging, we can think about automated report generation and assisting taking the notes from the doctor and analyzing the notes along with the medical images. Now what does it take, right? If you're working in AI in healthcare, what does it take in order to deploy your product into clinical practice, right? Let's see the steps. So the first and foremost step is identify a clinical problem statement which has a true clinical need. So this statement might be obvious. You might think that whatever problem statement you might pick has some kind of clinical relevance, but in medical imaging that's not true, because you might be thinking that it's a good machine learning statement, but it might pose more risks for the patient than the benefits. So it's always important that you analyze the risks versus benefits of the problem statement or the clinical utility you have chosen. And this is even as per the regulatory standards, you have to do it in the beginning rather than developing the product, because whatever you do in healthcare is time consuming and money taking. So once you are clear that whatever problem statement you have selected it has more benefits than risk, you can plan for the development. So like any kind of AI problem statement, the next step is getting data, right? So getting data, getting annotations for the data, and ensuring that there are not many biases in the data sets. So in medical imaging, all these three steps are difficult. One, we can't get variety of data because most often when we go and collect the data sets, we go to a hospital, and some diseases are demographic specific and regional specific. So you end up having data that is more focused in the sector where you are collecting. So always try to pool different data sources. Like if you're working on a problem statement, don't just stick with one data set. Try to pull data from as many data sets as possible. And the second critical step is authenticity of the labels. I think as Professor Yogesh sir has mentioned, right, the annotation of the traffic images. So here we can rely on the human labeling. But in medical imaging, we can't rely on the labels even by a doctor, because even the doctor's accuracy could be as low as 60-70%. Then how can we trust the labels given by doctor, right? So what we do in medical imaging typically is we try to employ more radiologists, take majority voting, so that we ensure that the labels what we are getting are more accurate and supported by majority of the radiologists. The other fashion is you take multiple tests. Like for example, if you're trying to detect cancer from X-ray images, then instead of just relying on the X-ray images, try to rely on other imaging modalities, maybe whether the person had biopsy or not. Take the biopsy results as a final ground truth. So because garbage in is garbage out. If when you are training, the labels are not good, whatever model you would be building in a real world scenario, it will not perform well. So always ensure, and when you are trying to work on any kind of open source data sets, ask the first question, how they are getting the ground truth, right? Many scenarios we don't typically look at it. We just take whatever the labels given in the open source data sets and then we try to analyze. But first and foremost thing we have to ask in medical imaging is how they are getting the ground truth. How many radiologists are involved? What is the main source of ground truth? Did they follow up? So these are the questions you have to ask if you are working in AI for medical imaging. The next step is anticipate potential biases, because there could be numerous biases which you can see in medical imaging. One example which I can give is there is a research lab. What they have done was they are trying to develop detection of breast cancer from ultrasound images. So they trained a model using a data that came from one hospital. They got very good results and they tried to deploy. And then the model failed. When they tried to look at why it failed, the reason they found is that whenever there is a cancer, the doctors are marking some annotation on the images. So rather than picking up the abnormality, it is just picking up those annotations and then telling whether it's a cancer or not. So that's the reason, like when it went to another hospital data, they're not doing this annotation and it is not picking up the cancers. So this could be one potential bias. What are the other potential biases? So if you're collecting data in a cancerous hospital and you're training a model from that, you are seeing already high risk patients. And now if you apply the same model on a screening population which you're doing in a screening camp, it might give elevated risk even for a normal patients. So you need to understand how we are getting the data, what type of data we are getting, so that we can minimize it. And imbalance is a common problem in medical imaging, because if you're talking about again cancer, you have to screen 100 to 1000 population in order to get 1 to 2 cancer patients. So imbalance is the main issue, and you have to come up with ways how you can balance it. Like you can upsample the samples or come up with some augmentation strategies for better learning. So once you curated the data, the next step is developing any kind of AI model. You can take any kind of off-the-shelf models or develop your own AI models. But one important thing is the models have to be explainable, because whatever AI models we are developing, we are giving it to a doctor. And if you simply say yes or no, they can't rely on the system. They need to know why the model is saying. So from the beginning itself, make the models more explainable and interpretable. And once you develop this, the next step is utilize appropriate performance metric. So we might think that accuracy is a good metric, but if you have 100 population and 99 are positive and one is cancerous case, even if you classify everyone as negative, you'll get 99% accuracy. So try to know what are the performance metrics that are more applicable to the problem statement. And estimate model utility and trade-off, which means you need to understand where the model works and where the model fails. This is very crucial in medical imaging, because we don't really require a system which works in all the cases. Even if it works in subset of categories, that itself is very good. I can give an example of mammography for breast cancer detection. Everyone knows that mammography doesn't work for dense tissue, but it is fine, because we know that if it is younger women or dense tissue, they don't recommend mammography. So trying to know where the model works or where the model fails is very crucial in medical imaging. Now, once this AI model development is done, the step which you won't see in other AI products and which you will see in healthcare is clinical trial and regulatory. This is a step which is a major hurdle for companies trying to deploy their products in clinical world. But this is a crucial step as well, because it ensures that whatever we're deploying are trustworthy and safe to patients. So in clinical trial, what happens is that whatever model we develop, we try to evaluate. How we can evaluate is whatever model we develop, we give it to a hospital and ask them to test it. And then we get the results, and using these results, we get the regulatory clearances. So typically, in order to do these clinical trials and these things, it takes a lot of time. So it's important you know that where the model works and where the model fails, because you can ensure that by setting these constraints, you can conduct the clinical trial properly. So once you get the regulatory clearance, the job is not done. You can deploy it, but the main thing is you need to ensure that doctors are really using it. So try to understand with doctors, whether they're comfortable with the model and how we can improve the clinical adaptability. Continuous monitoring and then updating the model so that it performs well is also very crucial. So just to make it interactive, so let's take a common hypothetical example. Let's say there is a researcher who wants to develop an AI system to classify pneumonia from PET scan. What the researcher has done is that he downloaded an open source data, and the open source data has 100,000 images that is coming from 1,000 persons obtained during COVID-19, and the labels are given by a radiologist. And what the researcher has done is they downloaded the images, all the 100,000 images, they split into 80-20, and then they trained a simple deep learning model, and they got 99% accuracy on validation set. And the researcher got excited and deployed in a real setting, and it didn't perform well. So can anyone tell or guess what could be the challenges or what are the pitfalls with this approach? Anyone? What do you think is wrong with this experimentation, where the researcher has downloaded 100,000 images of 1,000 persons who are during COVID-19, and they simply did 80-20 split, and then they classified whether the person is having pneumonia or not? Can anyone guess? Okay. So what is happening here is, for each patient we are having close to 100 images. That means same patient data is there in training as well as testing. So there is a training data leakage. That could be a reason why the model is performing very well. The researcher didn't check the ground truth veracity. It is labeled by a single radiologist, and we don't know how good is the radiologist. It could be that the radiologist is junior radiologist and the performance could be 60% only. there are many potential biases. We don't know what data sources, and it was collected in COVID-19 time, because during that time the pneumonia might be different because we have a COVID-19 pneumonia. So it could be that disease variant is different, like whatever worked in that population might not work on a simple pneumonia. So the researcher has claimed that he got 99% accuracy, but as we discussed, if there are 99 normal population and one cancerous or one pneumonia case, and you classify everyone as normal, you still get 99% accuracy. So the accuracy is not a reliable metric. You might have to see what is the sensitivity, what is the specificity, what is the AUC. There was no independent testing. It's always important that whenever we develop models in healthcare, try to do some kind of independent testing which doesn't have any link to the training data sets. And there was no risk benefit analysis. Is it really important to classify pneumonia using PET scan, right? Because PET CT typically uses radioactive tracers. It uses CT scan, which is ionizing radiation. Is it really important, or a simple X-ray is sufficient for pneumonia classification, right? So that kind of risk-benefit analysis was missing on this. So with this, I will move on to the next topic of how we are using AI to improve breast cancer detection. Why breast cancer, right? So if you see, according to the statistics, breast cancer is the leading cause of cancer deaths among women. It has both highest incidences and highest death rates in almost all the countries in the world. So in 2020 alone, even in 2022 statistics alone, more than 685,000 women lost their lives. If you see countries like United States, they have a good survival rates, like close to 80%. But in countries like India, we are losing every alternate woman diagnosed with breast cancer. Like we had a cancer incidences of 178,000, and you can see that almost 50% lost their lives. But breast cancer is one of the most curable cancers if we can detect early. If we detect the cancer in their early stages, that is more like as small as less than 1 centimeters, we can simply do a OPD surgery and then send the patients home. So it can be that curable. If it is stage 1, the survival rates are as high as 99%. But yet we are losing 50% of women to this disease. When we analyzed what are the main gaps in the current screening techniques, we found that X-ray mammography is the most common standard used for breast cancer screening. This has worked very well in countries like US in detecting cancers. But then we have to realize that whatever works outside might not work for countries like India, which looks at more affordable solutions. Talk about X-ray mammography, it is a gold standard outside India in high-income countries. The limitation is that it doesn't work for younger women or women with dense tissue. Because how it works is that we send X-ray beam and then we try to analyze tissue differences. Whenever there is a cancerous lesion that forms, it has a higher tissue density compared to the surrounding tissue. So that's how it works. But in countries like India, 50% of the cancer cases are happening below 45 years of age, which means mammography doesn't work for 50% of the cancerous cases for India. And unfortunately, even after 45 years, approximately 60 to 75% of Indian women or Asian women tend to have dense breast, unlike US women where this percentage of dense tissue is maybe some 40 to 50. But in India it is like 70 to 75% women tend to have dense tissue even after 45 years. So approximately 75% of the cases mammography is not applicable. So that could be a reason why we are not able to detect cancers in their early stages. And the other implementation challenges is the equipment costs close to 1 CR or 2 CR depending on the type of equipment we are taking. It is very costly, so it's very limited to metropolitan cities like Bangalore. And for both imaging and interpretation, we require high skilled expertise because we are using ionizing radiation, so there should be dedicated infrastructure, dedicated power supply, and the entire room should be sealed off to block the ionizing radiation. Ultrasound and MRI are recommended, but they are typically recommended as an adjunct to mammography. Like once mammography finds, ultrasound is recommended, or if the patient is already having some complaints, ultrasound is recommended. It has benefits of low cost and all these things, but the disadvantage is that we can't take it for screening camps because it can be used for fetal sex determination, due to which we can't directly go and then do a screening camp. Again, dedicated radiologist has to review these images for almost 20 to 30 minutes for each patient, which makes it less scalable. MRI is very, very costly, and it's typically used for high risk women. Due to this hand examination, we detect majority of the cancers with hand examination in India, and that is one of the reasons why we detect cancers in the late stage, because once we palpate, it's already stage 2, stage 3. So the treatment cost as well as the survival rate, survival rate reduces a lot, and the treatment cost increases if you're detecting in the late stages. So that's where we thought, can we come up with any imaging modality which could which could solve some of the problems which India or LMICs, low and middle income countries are facing. Then we found that there is this modality called thermography, which I will talk what it is. But you all might have seen during COVID-19 in airports or malls, you have seen these thermal devices which measure the temperature of the face region. It's the same thermal cameras. We just capture what is naturally emitted from the body, and then we try to interpret how, whether there is a cancerous activity or not. So the advantage is that it is very low cost. You can get thermal cameras as low as 60,000, 70,000. And you can see that that's a thermal camera. You can carry it in a backpack. It doesn't require any kind of sophisticated devices. It can be battery powered, so you can take and then do a screening in a rural camp. And the disadvantage is that it still requires manual expertise, which I will talk in details why it is a challenge. So what is thermal imaging? It is nothing but measure of surface temperature. Every body emits long wave infrared radiation. We just capture what is naturally being emitted from the body and then convert into temperature values. So whatever you are seeing here, each pixel is a temperature value. So since these are real numbers and we can't represent, what we try to do is we try to employ this pseudo color palettes, where the lowest temperature 19.8 is assigned to black and the highest temperature 35.5 is assigned to white. And then whatever is in between, we just linearly assign to these colors. So that's how we got this color image. So this color image might be helpful in interpreting these thermal images. So thermal imaging has been there since 1960s. The technology is found to have different applications, starting from cancer detection, not just breast cancer detection. It can be used for thyroid cancer detection. It can be used for oral cancer detection. It can be used for skin cancer detection. Diabetic foot ulcers. If you talk about diabetes, every year the number of people with diabetes are increasing, and unfortunately 25% of them are suffering with diabetic foot ulcers, due to which 10 to 20% of them has to go for amputation, like typically the amputation of the leg. So how do we solve that? We have to detect it in early stages. So the current techniques that are used in the hospital is like needle pricking or tuning fork mechanisms they try to use to understand. But there is no quantitative way of measuring the ulcers. Thermal imaging is very good because it can measure the pain. If the blood supply is stopped there, you can see a cold spot. If there is an inflammation, if there is an ulcer that's going to form, you can see a heat pattern. So you can measure these cold and hot spots to analyze whether there is any kind of foot abnormality that's going to come or not. Sports injuries is one area which, any kind of injury that happens, you would see inflammation, you can track. If there is a burn or anything that happens, again, how the wound is healing, we can measure, because if the wound is healing, you would see the inflammatory cells slowly reducing over time. So you would see the heat pattern reducing over time. Pain localization and veterinary medicine. Like unlike humans, animals can't express this pain. So how do you know that where there is a pain? So that thermal imaging can pinpoint where exactly is the inflammation happening. Now, what is the challenge with medical imaging? Now, whatever image you are seeing, right, as we discussed, if you see the image on the left hand side, the 25 degrees is assigned to black and 39 degrees is assigned to white. But let's say another radiologist has assigned 28 degrees to black and 36 degrees to white. Both are same temperature values. Temperature values are same, but we adjusted the temperature limits, due to which the entire image has changed. Whatever you are seeing completely normal, like green color region, now became red color region. And in fact, this is a IDC cancer, stage 4 cancer. So it is highly subjective for the radiologist. They require a lot of expertise in order to adjust these temperature palettes and also to analyze different colors in the image. And also not all high temperature are due to malignancies. It could be for benign conditions as well. So that's where we worked on this novel integration of artificial intelligence and thermal imaging to make the interpretation easier and more accurate for the radiologist. So what happens in when there is a cancer, right? Cancer is nothing but uncontrolled cellular growth. So the cell starts multiplying itself. Now, in order to do that, it requires more resources. So it basically draws more blood supply, it forms new blood vessels, it pumps more blood from the existing vessels. All these increase the heat pattern around the tumor region and also it increases the vascular activity. Now we have thermal cameras which can detect temperature values up to 0.05 degrees centigrade, which we can't even detect with eye. So those can be analyzed and then we can use AI algorithms in order to understand these minute patterns and then make it more interpretable. Generate an image automatically from this more red or orangish region, what you are seeing, to a more clear image for the radiologist with annotation so that they know that this is the malignant lesion. So in short, instead of looking for lumps with the help of AI, and so instead of looking for lumps means because in India we are still using hand examinations where we are trying to find whether it's a palpable lump or not. So instead of looking for lumps, now with the help of AI and thermal imaging, we are trying to look for cancerous activities. So which would help us in detecting cancers in their early stage, thereby saving lives, also reducing the treatment costs. So this is the entire screening setup. So one of the beauty of this thermalytics is that it's completely privacy aware. Whatever imaging techniques which we have discussed, in all those things, the woman has to undress in front of a technician for screening. Whereas in this thermalytics, we ensured that the entire screening process is privacy aware. The technician is outside, woman goes inside a room. It can be with a curtain cloth or it's more like a in a shopping mall changing room kind of thing. The woman goes inside, sits on a stool, and we have a thermal camera, and there is a cooler to blow air so that if they're wearing tight dress, if they're coming from hot environment, they basically attain equilibrium with respect to the surroundings. And the technician completely operates from outside. So it is non-contact, non-invasive and privacy aware. There is no radiation because whatever is naturally emitted from the body, we are just collecting the heat pattern. It works for women of all ages and even men too. Even men can get breast cancer, and 1 in 1,000 men has a risk of getting breast cancer. And there is no imaging modality for men. So thermal imaging is showing potential to be a good imaging modality there as well. It is affordable. As we are seeing, we can carry this in a backpack and then do the screening camps in the rural areas, so that specifically where we are not able to provide healthcare now, with the help of this technology, we can go and then do the screening. and it can detect cancer much before a lump is felt. We have studies that shows that we can detect cancers as small as 4 mm lesion size. So what is the overall procedure? It is a simple 15 minutes test. Whenever participant or whenever participating woman comes, the technician explains about the test. The next step is cooling the subject for 10 to 12 minutes so that they get stabilized with the surroundings. We capture 5 images because a single view might not capture the entire 180 degree of the breast heat pattern. We capture 5 views at 5 different view angles. Once that is done, we upload this into Thermolytic software, which automatically generates a report, and it sends to the radiologist. And once the radiologist looks at it, certifies it, it comes back to the technician who does the imaging, who can download the images and then give it to the participant. It's a simple 15 minutes test that we can do. But in order to generate this report, it's not often a single machine learning model that we see. We have bunch of AI modules to make this reporting accurate, interpretable and explainable to the doctor. So if you see at high level, the first and foremost thing is automated image quality. So in medical imaging, the quality checks is very critical because, as we discussed, garbage in is garbage out. If you don't give high quality images, the predictions might be wrong. If the predictions are wrong, it again, it could lead to false positives for the patient or false negatives, incorrect reporting for the patient, which is not good. So we want to ensure high image quality. Then we have modules which automatically identify the regions of interest. So in this scenario, breast is a region of interest where we want to identify the cancers. And once we do that, we try to identify potential features what the doctors look for. Typically what doctors look for is that they look for any kind of high regional activities, which we call it as hotspot. So we automatically identify that so that doctor, instead of identifying where the hotspot is, we already give them, okay, this is the hotspot, and we give quantitative radiomic features out of it. Similarly, they look for vascular activities. So we automatically identify the vascularity and then give the vessel radiomics. They look for any kind of areolar asymmetries. So we try to get the areolar hotspots and then extract areolar radiomics. So this way, we are just mimicking what radiologist is doing. So that way it is more friendly with the radiologist. Instead of spending so much time on the images, they can just rely on these parameters and give a quicker and more confident decision. So just to go into details, right? So the first one here is we want to just convert this image to a contrast enhanced image, right? And for this image quality checks, we had 3 main quality checks that we want to look at. When we capture the image, we are capturing 5 images. One of the common mistake technicians does is when they are capturing left image, they often label it as right image, because when they are capturing the left breast, it's on the right hand side. So they label it as a right image. And now the model, instead of saying the cancer is in left breast, it says cancer is in right breast. So which changes the entire clinical flow. So in order to rectify or correct these mistakes, right, we have a deep learning algorithm which automatically, given the image, it can predict and then tells the technician, hey, this looks like a left image, do you want to proceed or not? So and it automatically corrects it. The other common mistakes which we see is they might not cool the patient well, because they might be wearing tight dress or they might be coming from the hot environment. If you don't cool well, then we might see strong heat patterns because of the heat residue, because of the ornaments or because of the dress itself. So those can come as false positives. So we have algorithms that can identify whether it's cooled properly or not. Similarly, focus check, because we are relying on the temperature values that we are getting. If the camera is not set properly or the camera focus is not set properly, the pixels get averaged out. So the temperature gets averaged out. If there is a malignant lesion, we might see a lower temperature because of this averaging. So we have a focus check which can automatically identify whether it's a focused or not. So the second step is identifying where the breast tissue, because we are talking about the cancer in the breast region and we are capturing the images from neck to abdomen. Now the challenge is how do we capture, how do we segment the breast tissue? And this is a little bit hard problem because unlike rigid object where we know what exactly is the boundary, if I ask you where is the upper boundary here, you might say this could be the boundary or this could be the boundary as well. There is no single answer for it. So that is why even when we asked 2 radiologists to mark this breast region, and when we compared their accuracy between them, inter-dice correlation, that itself is 91%. That means only 90% of the region around that is matching. So which also tells that we can't get a 100% ground truth in this scenario, because even the ground truth itself is variable. We trained a simple mobile net backend based VNet architecture, which is an encoder decoder architecture, and we are able to get close to 92% dice index, which is close to what even a human person can get, because the inter-dice correlation is 91%. And now the second step is, once we get the breast tissue, how do we classify? So we employed a radiomics for classification. What radiomics are nothing but it's just radiological features which are more explainable for the radiologist, because the radiologist is taking a decision whether there is a cancer or not. If you simply give yes or no, ultimately they will not be able to rely. They need to know why this is cancerous, where is it. So we try to go into the semantics of it. So we try to annotate the images with the hotspots, areolar, vascular regions, try to give the radiomic features which could help them in a cautious decision. So you can see the report. Now the first thing what you see is these annotations what you are seeing here. So this will immediately tell the radiologist that okay, these are the abnormal regions that the model is looking at, and because of this, what is the likelihood of malignancy? The radiologist can get that okay, 80% chance that because of these regions that we are seeing, it is malignant. We are giving the vascular activity in the breast region, using which the algorithm is seeing that it's 52% likelihood of malignancy. And then again, areolar score, around the areolar heat patterns, what is the likelihood? So what happens is whatever radiologist is mimicking, radiologist is viewing, we're trying to mimic in the report. And each of these things are again explainable in terms of English format as well as the individual radiomic features. So in order to evaluate, we have conducted multiple clinical studies, and till now we have done close to 250,000 women in India alone, and now we are in 20 countries. And when we compared with mammography, if you see the results, we screened 769 women, and mammography we are able to do for only 404 patients, because approximately 1/3 of the participants couldn't go for mammography because they're younger women or they have some kind of pain or some kind of abnormality. Even in this cohort, the sensitivity is 100% and 66.7% for mammography. So you can see a good improvement in the sensitivity, and it can work for majority of the women. And as we discussed, the next step is to get the regulatory clearances. And if you're interested to know more about how these technologies work, so you can look at these papers, and if you have any questions, you can feel free to write me as well. Happy to take any questions. Yeah.
Thank you, sir. I would like to thank Dr. Shivateja Kakileti to his remarkable research contribution in the field of medicine. And I would like to give a brief introduction about sir. Dr. Shivateja Kakileti, a distinguished principal research scientist and director at Niramai Health Analytics, renowned for his pioneering work in artificial intelligence and machine learning for medical imaging. As a key contributor to the development of radiation-free and non-invasive breast cancer detection methods, he has made significant strides in healthcare innovation. A PhD holder in medical imaging from Maastricht University, the Netherlands, and a BTech graduate from IIT Guwahati, Mr. Kakileti has co-invented 25 international patents, authored over 30 publications, and contributed to 3 book chapters. His remarkable contributions to AI-driven healthcare have earned him prestigious recognitions, including Forbes India 30 under 30, Forbes Asia 30 under 30, and BW Healthcare World 30 under 30. He has also been a recipient of several fellowships, such as the EBCC 2022 fellowships, UKIO Bursary 2023 and DAAD Vice Fellowship 2014. With his deep expertise and unwavering commitment to the advancing medical technology, Dr. Kakileti continues to revolutionize early cancer detection, making screenings more accessible and efficient. We are privileged to have him with us today and eagerly look forward, and we have had a great session with sir. Thank you, sir. Now I would like to welcome Dr. Babita ma'am to give him her honor of.
Solar energy to drive up the mechanism to decompose the organic pollutants. This technique is also a greenhouse technique because of the simplicity and cost effectiveness, mild reaction condition and low energy consumption. And there is a use of molecular oxygen as an oxidant material without the addition of any chemical additives. So the drawback of this method, this is a traditional method, so the drawback of this method is a trial and error process. So by affecting the factors like optimizing factors like the pH, the concentrations, they are affecting to it. So for that purpose, the AI came into existence, which optimizes the factors like which are used for the potential photocatalysis with the required properties. The role of this AI will accelerate the photocatalysis by enhancing the catalytic design, like the compositions of the catalyst and the nanostructured material, and the modeling of the reaction mechanism, the pathway in which the reaction is going to be taken place, and the optimization process, which includes the catalytic stability, scaling up the photocatalytic process. Now, intensifying the photocatalytic performance by the defect and doping engineering, which will, and this AI will help us to analyze that what are the factors that are affecting the material properties by guiding the analyst to fine-tune the photocatalyst. Now, AI-based understanding reaction mechanism, the computational models help us to predict that the reaction pathway and the mechanism which are used to degrade the organic pollutants and enhance the photostability and its efficiency. The molecular visual representation helps us to interact how the reactant and the photocatalyst are interacting with each other to degrade and to decompose the organic pollutants. Now, generative AI for novel material discovery. These variation autoencoders, generative adversarial networks, these are the techniques.
Highlighted these benefits, right? So first, photocatalysis means you are using light to catalyze the reaction, and you are looking at some environmental degradation in the form of semiconducting materials. So your AI, what is it actually doing there?
It is helping to it is optimizing the factors like in
Tell me 5 factors that is important in photocatalysis, which you can now model via AI and make it useful.
Yes, sir. In traditional methods of
not only about traditional.
Yeah, in that only. So it is a trial and error method. No, we are keep on changing the concentration and maintaining the semiconductor material.
Okay.
So this is a trial and error process which leads to a time consuming and the wastage of material. But this AI tool, what it will do?
So what will you give as input to your AI?
Yeah, it will like the conditions. What are the conditions? At what concentration we have to put the at what concentration it is required to carry out the efficiency of.
That is what we should discuss as a case study.
Yes.
You got it now. So when you discuss the case study, you only point out now. So these are the parameters now that is traditionally done. Okay, these are the various concentrations or the trial and error methods or whatever approximations we do. So this is the data set that is available. Now I am doing SVM or random forest or whatever it is, alright, using all these algorithms to come up with simulations which will help me do photocatalysis better. So there is a case study that has been done where the efficiency is 99% done. Job done. Got it? So if you strike on that, then your presentation would have been wonderful. But now it is good. All the best.
Of the zooplankton diversity and water quality analysis in Alasandra Lake, located in Yelahanka, Bangalore. When the zooplanktons, they are considered to be a bioindicators which helps to indicate the water quality, and because of their presence we can analyze the what is the water quality present, and they are the good indicators of the climatic change and water quality, because they strongly affects the environmental conditions and responds to the change which takes place in the environment. These zooplankton community, which plays an intermediate link between the secondary phytoplanktons and the fishes, and they form an intermediate link between fishes and phytoplanktons. Our objective of this presentation is to investigate the different types of zooplanktons which are present in that Alasandra Lake, and to analyze the water quality parameters like BOD, dissolved oxygen, chlorinity, salinity, and the organic matters, and to access the correlation between the zooplankton diversity and the pollution level which takes place in that lake. We use this study design into like qualitative analysis and quantitative analysis. Like qualitative analysis, we used to collect the water sample, surface water, and we used to store that water in 4% formaldehyde, and these zooplanktons have been observed from by using the microscope. In coming to the quantitative analysis, there we used to collect some water sample from the lake, and we used to measure the dissolved oxygen level and biochemical like BOD test, salinity, chlorinity test, and the organic matter by using these Winkler's method, volumetric titration methods, and Moore's method, and KMNO4, H2SO4. These methods we are using for analysis of quantitative zooplankton analysis. Coming to the result, we have analyzed about 10 months. In these 10 months, we are able to analyze the zooplanktons diversity present in the Alasandra Lake. Like during the Jan, Feb, and March, during summer or winter season, the zooplanktons rate will be very less, and during the monsoon season, like rainy season, the zooplankton diversity will be more, and with respect to the climatic conditions, the presence of zooplanktons will also be increased in the lake.
So with this analysis, what is that you want to do in the domain of AI, for example? How you want to know? What you have done is what you have physically your project work activity is what you have presented, I suppose. So this theme of the event being AI. So what is that you want to now try to do with respect to using any of those models of prediction? Is there a predictive approach that you can now follow? Can you now get all the data, 10-year data, and then make it into an AI model? Have you thought about it?
ಏನು ಮಾಡಲಿಕ್ಕೆ ಅದು ಏನು ಅನಾಲಿಸಿಸ್ ಮಾಡಿದಾರಲ್ಲ, ಸೊ ವಾಟ್ ಯು ವಾಂಟ್ ಟು ಶೋಕೇಸ್ ಲೈಕ್ ಎಕ್ಸಾಕ್ಟ್ಲಿ ಏನು ಹೇಳಕ್ಕೆ ಹೊರಟಿದ್ದಾರೆ?
Different types of planktons present in the lake, and how the number of planktons vary from season to season. That we are
how it affects the fishes and other. So fish population ಏನಾಗುತ್ತೆ, for example, October.
Fish population.
For example, go back to that chart.
You said the rainy season and it will be more.
So example, June, July. Okay. So what will be the impact on, say, fishes?
So the oxygen level will be more in the lake. So due to this, the zooplanktons will be abundance in the water. So
how will affect the fishes, for example? That is what is the.
It increases the fish growth.
You were able to correlate that? How much? By how much was it increasing? For example, Meenugarike is help. For example, to help in fisheries, your project will become very valuable, right? So, after they correlate part, because there is a product that people are looking at, right? So, they want to do enough of fishing. So, you can now alter it. And even in the month of January, February, you can now try to do something. In order to maintain the fish equilibrium or that quantity, then you see your work will have national level value. Correct? So, think about it.
Quantitative analysis model in 98 of the real parameters.
Our topic is about assessing the relationship between organic carbon, texture, soil moisture and land use in different areas of rural and urban Bengaluru. The soil organic carbon content is a key and plays a critical role in the fertility of the soil and also the agricultural ability. The soil organic carbon depends upon the precipitation and temperature of the land area, which has a direct link between the soil moisture and also the texture of the soil. The main objective of our study is determination of pH, temperature, soil texture, soil organic carbon content in different land uses of urban and rural Bengaluru, and also to study the influence of this SOC on soil texture as well as the moisture. The materials and methods which we use includes the soil sample collection was done by a soil collecting probe which was inserted at the 10 cm depth of the soil, and 6 samples were collected from each different lands. And for the determination of SOC, we used Bagley-Black method, which is a wet oxidation technique. For determination of soil texture, we use texture triangle method. For moisture and pH, we use soil tester device, and for temperature, it was thermometer. From this, we got the result of soil texture, which has from the farmland of areca and corn, we got to know that the soil texture varied from clay to silt loam type of soil. In barren field and playground, we got sandy loam to loomy sand type of soil. In garden and park, we got clay type of soil. In lakes such as Alachala and Tutanali, we got silt loam to clay loam type of soils. The soil parameters which we worked on includes soil carbon concentration, which was measured at mg per gram, where the areca farmland, park and garden and lakes had higher carbon concentration than that of barren field and playground. Same goes for moisture, which has similar type of graph where the vegetative lands have higher moisture content, whereas the non-vegetative land have lower moisture content. And in pH also, the barren field and playground, which has low moisture, also have the slightly acidic type of pH, but the vegetative lands has alkaline type of pH. And even in the temperature, it has opposite impact, where the temperature of barren field and playground, where the moisture is nearly 0%, has higher temperature value. From this, we can say that the soil organic carbon content varies with the moisture and also the texture of the soil, where in the cornfield and areca farmland, the moisture was very high and the texture of the soil was from clay to silt loam type of soil. From this, we can see that the vegetative land have higher carbon content. In also the garden and park, which has a clay type of soil, has higher moisture retention, which enhances the carbon content of that soil. In lakes, as it is near the lake soil,
Technical session for Dr. Saujanya ma'am, Senior Principal Scientist, CSIR, IICT, Hyderabad. Ma'am, you can take. Now I request Professor Shambhavi ma'am to introduce Madam.
Good afternoon, one and all. I, Shamali S., take an immense pleasure to welcome Dr. Y. Saujanya, a distinguished principal scientist at the CSIR, Indian Institute of Chemical Technology, Hyderabad. With an extensive academic background, she earned her BSc and MSc from Sri Venkateshwara University, Tirupati, and PhD from the University of Hyderabad. She further honed her expertise through postdoctoral research in France and Canada before joining IICT in 2004. The research work mainly focused on computational chemistry with application in organic chemistry, bioorganic chemistry, and material science. She has made a significant contribution to the design of material for solar cell applications, eco-friendly carbon dioxide absorption materials, and computational modeling of chemical processes. Her work is widely recognized with over 50 publications in reputed international journals, and she has led multiple sponsored research projects by DST, BRNS, and other national agencies. In addition to the research contribution, ma'am is an active mentor guiding young researchers in the field of computational chemistry and material science innovation. Her dedication to advancing the science and sustainability has earned her a strong reputation in the scientific community. We are honored to have you here, ma'am, and eagerly looking forward for your contribution. We welcome you, ma'am.
Good evening to one and all. I think you are all got exhausted, right, with a series of poster and paper presentation. I try to be more informal and try to get connected to you through my discussion on AI and ML for materials discovery. So you can just interrupt in between, because this session I want to be very informal. Not like whatever slides I have, I don't want to force you to listen to everything. You can just ask me questions in between. Some AI and ML sessions in the morning, right? So you are all aware of fundamentals of artificial intelligence and machine learning? So I don't need to go in detail what is AI and what is ML and what is supervised learning, unsupervised, and so on, right? So I hope you are all aware, because you can save time. I'll just go very focused on the how we have used these methods in identifying the very potential materials for our application. So before it gets started, any one of you, can you just tell me what is the difference between artificial intelligence and machine learning? Or both are same? Both are not same? Anyone, you can just get up and then try to see, because they are not same but connected to each other. Can you give an example of AI and machine learning? Exactly. Very good. Very good. Nice to listen to that. So, like AI, machine learning is a subset of artificial intelligence. Like the driverless car all these all automated models where human intelligence is required is AI, whereas machine learning depends on algorithm where data is required. So machine learning is a subset of artificial intelligence, and also nowadays in recent development we have deep neural network and GPI and so on. So but for most of our automation where artificial intelligence model is very much successful, for scientific purpose it is the machine learning which is more important. Why it is important? I will just give an example. Do you know when this AI and ML has come into existence? Is it in last decade or it is known for a long time? For a long time, right? But now why there is more buzz about it now in recent times? Because there is a lot of data has been generated. Why data has been generated? Because we have a very advanced computational hardware. Infrastructure has been very much developed in these days, so that we are getting lot of data in all the domains, right? You might be knowing this data in terms of health, in terms of finance, business, all the e-commerce. So all this is related to data, right? Likewise, how the data is for a scientific discipline like chemistry, biology and botany, and also for material science, right? But how the methods we use to develop models for the business models or finance models or e-commerce model is completely different in handling the data which is related to scientific disciplines. I will tell you why. So to develop a machine learning, I will focus more into machine learning models because this is the most appropriate model to work on a scientifically related data. And how the data is, how can we get the data for a scientific purpose, like for any for biological purpose or for identifying a material for a battery, for a semiconductor. So can you just give example how can we get the data for this kind of research? Like you want to have a semiconductor, okay? So how do we get the data? For otherwise you want to know the electrode that is being used for a hydrogen fuel cell, which is actually used in all electric cars and electric bikes and all. So to have a material for an application, we need data. Can you just give me how to get this data? Can you just suggest me where are the sources for getting the data related to this discipline, whatever I have mentioned now? How can we get the data which is related to materials discovery? I know you are all aware of it. Have you heard of PDB, Protein Data Bank? Protein Data Bank. So where you get all the crystal structures of proteins, right? This is for biology. Likewise, have you heard of all crystal structures of organic molecules, inorganic compounds and all? There are several databases available online where several researchers have worked on identifying these materials, and they have deposited the structural and geometrical, electrical, optical properties of these materials into database. So we have access to this kind of data. Okay. Just since slide is open now, I'll tell you very briefly from where I am coming. I am from Indian Institute of Chemical Technology, located in Hyderabad. Can you switch off this light so that the glare will not be there? I don't think people at the back can see it. Yes. So, have you heard of CSIR, Council of Scientific and Industrial Research? It comes under Ministry of Science and Technology. Under this umbrella, there are 37 national labs located across India, various places. So, across India. So, in Bengaluru, we have National Aeronautics Limited, that is aerospace related chemical lab, and also we have 4th Paradigm Institute. In Hyderabad, we have 3 CSIR labs, that is CCMB, NGRI and IICT. I am coming from Indian Institute of Chemical Technology. It focuses more into chemical sciences. Likewise, in NIL in Bengaluru, it works on aerospace work, aerospace engineering. And likewise, NGRI for geophysical research, and whatever earthquake we experience across India, the seismic data will be generated in this lab. Likewise, we are having 37 national labs, and Indian Institute of Chemical Technology, we actually inaugurated in 1944, so we completed 80 years of our service to the nation, and our institute contribution spans different domains, health, like strategic sector, and agrochemicals, and pharmaceuticals, etc. So, very briefly I will tell you what we have in recent years. Have you heard of Covaxin? Yeah, Covaxin, we have developed the process technology for a molecule called adjuvant, which stabilizes the vaccine when we inject the vaccine into our human body. That process has been developed at IICT. Likewise, we have waste management process technology, and also we have developed process technology for several active pharmaceutical ingredients, which we supply for several clients across India. This is briefly about our institute. So, if anyone is interested to see more about our work, you can visit our website iict.res.in, and we also conduct skill development program in several research domains. If someone is interested, you can apply, where we can provide you the accommodation and other logistics. So, very briefly, we have 4 paradigms of science. Like in human scientific human evolution, in the beginning, how the science has developed, like just by observing, the planetary motion, Earth orbit. So, by looking at that, the Galileo, Kepler law and other astronomical observations have been arrived, right? This is called the first paradigm. The second paradigm is based on the theoretical assumptions, that is Newton's law, classical laws and all. So, the third paradigm is based on computational simulations, where the computational architecture has been used to simulate the observation, the hypothesis that has been put forward in the first and second paradigm. The fourth paradigm is called the data-driven paradigm. As I mentioned earlier, because of so much of data has been generated, we have the fourth data-driven scientific evolution. Likewise, we are into fifth paradigm now, where AI and ML are the scientific revolutions. So, these are examples of AI. I don't want to get into that. So, we have AI and ML in every field. What I said, e-commerce, finance, business, health. So, how it is for the material science? So, why do we need new materials? In human evolution, we see the advancement in materials discovery from Stone Age, from Iron Age, Bronze Age, where the new materials has been discovered because for our comforts and for our needs, right? In every age there has been some other or other materials have been discovered. So here now, what is the main material that is being used now? Can anyone give an example? What is the very like Stone Age, we have iron, bronze and all, right? Silica, exactly. It is the silica age, and also the plastic. Plastic also is a correct answer. So likewise, there is always in human progress, we are curious to know more application, more properties for a materials discovery. So some materials are discovered because of their mistake, because of some accident, like X-rays, right? It has been discovered by a chance. Likewise, what we use for car and other vehicles, the rubber, it has also been discovered by an accident. So those are some very few examples that has been discovered. But what is the traditional approach? Traditional approach is, before I will go into that, why do we need materials in the current age? What are the challenges in the current age is to have a good environment, pollution, toxic free environment. All major cities are polluted, right? So we need to have a material that can take, that can absorb, that can remove the toxic gases from the atmosphere. And also we need to have a material that can run in our batteries for electric vehicles. Likewise, we have, these are called our challenges, the sustainable challenges that we need to look for the materials discovery. So, that coming back to that one, what is the traditional approach for having, because we always look for a new material with improved properties, with resistance to the high temperatures, whatever the conditions we are using. So we always look for a new materials. What is the traditional approach? We look into, if you just, you know this one, right? Periodic table. So each element is a very potential for a material discovery. So if you want to scan this material which is based on several combination, you will have n number of combination. Can anyone make synthesis of these combination, do the characterization, testing? Is practically impossible, right? To have a one material to have a very good properties, it is traditionally very tedious and very laborious process. So that is based on the fundamental chemical and physics laws. So whereas the empirical, which is, as I said, because of huge amount of data that has been generated, it is more into the empirical methods. So the time required for the synthesis of one material will take a PhD time. Likewise, it takes even for simulation to do I said in the beginning, in the third paradigm of science, computational simulations have been used to discover new materials, right? Likewise, to do the computational simulation for a single combination of material, it also will take a huge time. So because of the high performance computing, that is infrastructure that has been we are enjoying, if you merge with data driven AI and ML modeling, we can significantly accelerate the discovery of new materials. So we are combining the computational hardware that has been upgraded or developed in recent times, along with the data that has been stored in several resources to discover a new material in a faster way. So this is very important information here. I think this is useful for every one of you to understand the time length and the time scale and the length scale and the accuracy. Like you have a problem of a material like a glass which is composed of silica. Okay, so if you go to a biological molecule like a protein or enzyme, the length of it is much larger than the silica, right? So likewise, you have a problem which will span different length scale. That is, the size of your material is from picometer to angstroms to some millimeter. So if you want to select a computational simulation model, there are different methods that has been shown here. This is the outline of the different methods that has been used for different length scale and the time scales. If you want to work on a silica atomic model, you need to apply these quantum mechanical methods. If you want to work on protein kind of system, you have to work on this kind of dynamics. This is called molecular dynamics. So there are several range of methods that we have to choose depending upon our length, depending upon the problem which we are looking into that. Accordingly, the accuracy also will change. For example, if you work on a simple atomic model, the accuracy is very high. Whereas if you increase the time length and the time scale, accuracy also will reduce. So what I want to tell you, to make you to understand, is there is a trade-off, balance between the problem which you are working and also the accuracy which you want to work on it. So this is the one which I have explained you, that there are several in traditional way of discovering materials, which is very laborious and tedious to discover new materials. So okay, now the machine learning, artificial intelligence, data driven models, these are all buzzwords and we can use it for discovery of materials and so on. This is very attractive to listen, very fancy and all, right? But there is always challenges. For everything we have to cross these barriers. What is the barrier? Like for health sector, if you want to work on machine learning or AI model, what is the data? If you have a patient data, like if MRI scan data or abdomen scan or anything that is related to disease, if you have an images or patient data, you can work on this data and develop a machine learning model, which is very available across our hospitals, right? Only the problem there is the data is not properly digitalized. That is the only problem. Here, when you come to the discovery of materials, so I asked in the earlier slide, what is the source of the data? I think someone has answered, right? How do we get the data? Have you heard of publication? Publications in journals, right? Can you give any example of any scientific journals? Whatever the research work you are doing or in whatever subject you are very much familiar. Any journal name? Nature, exactly, very good. Nature, Scientific Direct, Science, and American Chemical Society, likewise Physical Society. Have you heard of Wiley, Elsevier? These are all publications, publication houses which publish our work. Like whoever the scientists who are working at different institute, when after they identify some results from their work, they want to publish in an international journal. So in this way, we get access to this data that is being published in these journals, either in physics, biology, chemistry. You can just Google what are the journals available in the subject which is of interest for you. Then it will give the list of journals. So if you open any journal issue, you will get lot of papers that has been published in several domains. So if you open any journal publication, you can get lot of data, lot of graphs, lot of tabular data which is shown here. So since as I said, I always look for the materials discovery. I will try to collect the data that has been published in these journals and also from the databases. There are databases available online. So I'll try to download this data and then try to see how they are compiled. Okay, so I'll just quickly complete whatever I just want to make you to understand. So in machine learning, what is a important factor is the data. Another thing is what is the quality of this data. All the data cannot be used to develop a model. So it has to be highly quality data. What is meant by quality data means the data has to be normalized and it should be reported at the same temporary conditions, experimental condition, and there are several there are attributes which the data has to be confined. Likewise, there are for example here in have you heard of unsupervised learning and supervised learning? I don't need to give examples, right? So what we do in materials discovery using machine learning is we take reported data and then try to, using algorithms that are available, we will try to make the computer to train based on the data which we give it to the computer to arrive at some patterns. For example, in this college we have suppose we have about 2000 data points. We have 2000 data points related to students and faculty. Okay, so now we have to categorize into different gender, the class they are taking, and other designation, subject and all we can classify, right? So based on these parameters, we will train the computer to arrive at some model. Likewise, for several compositions of the data, elemental composition that are available in the periodic table, we will train the computer to arrive at some model, so that we use this trained model to predict the properties of a new material. So the success of this model will depend upon the data that we have given it to the computer for training purpose. So that is the main thing one has to understood. So if you have any doubt, you can ask at this moment, because that is very important concept of machine learning for the discovery of materials. You need the data, you need the quality data. Okay, that is the first point. And the second point is you are going to train the model, you are going to train the computer based on this training set, that is available data, either it can be experimental data or simulated data, and they try to arrive at some pattern, like how many for example, for composition based on silica, oxygen, and silica and oxygen are the main composition for the glass, right? So based on the ratio of silica and oxygen, what is the density, what is the viscosity of the material, what is the toughness of the glass. So if we have this data and we give it to computer to recognize the pattern, it will arrive at some model. Okay, so that is called the trained model. Computer has understood something related to composition, the ratio of silica and oxygen related to the density or viscosity of that material based on the data we have given. Using this model, we will try to give the data, unknown data, which we don't know. We will give the element like carbon and oxygen. We will ask the ML model, okay, you know what is the composition of silica and oxygen, you know how the property, function of it. But with the unknown material composition which I am giving it to computer, can you predict the property? So that the computer will, that algorithm, computer in my sense is the algorithm, will predict some property which we are interested, like density or viscosity or any other property based on the trained model which we have given it to the computer. So consider, so this is the 3 important parameters for developing a model. This is the data set of materials which we have to have, either it can be composition, manufacturing condition and density, and also we need to have a relationship between this composition, like silica and ratio composition, to have how tough it is the glass, and also the density of new and untested materials. So based on this model we will predict the properties of unknown materials. So this is very fundamental concept, how the model will work on the data based on either it can be atomic model or it is a molecular based model, or it is based on the macromolecular data. So, so machine learning, the success of the machine learning depends on the parameters which are either the product based and also the manufacturing based. Likewise, like the product based means the output of this the material, the function, the toughness, the density of a glass, that is the product function. Whereas if you want to have a manufacture this material at the large scale, tons of glass you want to manufacturing. For that manufacture attributes, you should have a relationship between these two to arrive at very successful ML model. So coming back, what we are doing at Indian Institute of Chemical Technology, it is an interdisciplinary team, like AI ML experts who are based on computational science. They can develop some models to work on the data, but they will not have a knowledge which is biology based or physics based or chemistry based. So it should be an interdisciplinary team which has to work together to work on any problem or any project. So these are the databases which we generate the data or we collect the data, and as I said, these are the journals. These are the journals where you get lot of data related to whatever problem you are looking at, wireless, Springer, ACS, APS, and from the data we collect, we do this exploratory analysis, like all this the workflow to identify the potential material compositions. So this is an example which we worked on, water splitting for hydrogen production. So this is, if the experimental data is not available from these databases, what should we do? We look into the computationally simulated model data. That also that is based on quantum mechanics. So that also we generate in absence of any experimental data. How do we generate the data? This is the workflow we follow to get and then identify the patterns and then validate the training set model and then do the experimental testing. So what are the main areas we are looking? As I said, even the Bengaluru, Hyderabad is there is so much of pollution because of CO2 emissions, right? So there is a projects there which are funded by the central government to work on materials discovery to remove these gases from the atmosphere. So most of you are using bikes and automobiles, car and all, right? So there is lot of CO2 emissions that has been generated to the atmosphere. So it is very much diffused, all these toxic gases. So how do we remove? We need a material. How do we discover? Can we discover based on experimentals of all these materials step by step? It will take millions of years. So we are following data-driven model where we have collected the data from different resources and identified the relation between the data composition and its properties. Like the properties means function. And likewise, we have worked for a material discovery for the removal of carbon dioxide from the atmosphere. So how do we? This is the graphical representation how we have collected the materials data from the different resources. So it will take a minimum of 6 to 10 months to collect the data, and then we clean the data, process the data. These are all identify this. This is the pattern we have identified. What are the important attributes which are responsible, and then we developed a new composition for the material, and then we also did the computational model, and then from the successful identification from the machine learning, we did the experimental synthesis of the lead composition and then tested in our lab to get the very good performance. So this is the one which we have collected, and this is the softwares and other programs we use in our lab. So this is our team working. It is interdisciplinary team working on our projects, and I thank you for your attention.
Vice Principal Dr. Shamishta ma'am.
To thank everyone. So ladies and gentlemen, esteemed guests, fellow delegates on the dais and off the dais, it's my pleasure to bring this conference to a closure. So Maharani Lakshmi Ammani College for Women Autonomous organized Samagatha 2025 in collaboration with KSCST, that is Karnataka State Council for Science and Technology, and in alliance with Viksit Bharat, that is on 25th and 26th March. So this year's theme, Artificial Intelligence, Technologies and Innovations for Sustainable Future, paved the pathway for much needed disclosure on one of the most contemporary issues the world faces today. Dr. Vijay UT, the Executive Secretary of KCST, served as the chief guest, and Professor K.B.R. Verma Sir, Trustee MLAC, graced the occasion as guest of honor. So Samagatha 2025 layered fun with work through an intercollegiate science fest on the first day, that was on 25th March 2025. Events like crossword, treasure hunt, quizzes, dumb charades and connections sent students jostling and packing for the entire of the day one. The second day amazed curiosity and inquisition with various facets and nuisance of artificial intelligence brought to light by notable speakers like Professor Saudamini Ma'am from NCBS, Mr. Shivateja Kakiletli from Nirmay Health Analytics, Professor Yogesh Simhan from IAC, and Dr. Saujanya Ma'am from CSIR ICCT. The day was also marked by paper and oral and poster presentations where students, researchers, young and experienced students put forth points of inquiry and exposition. The paper presentation was in both hybrid mode, enabling the participants to participate from all over the India. The proceedings of the abstracts have been released by our chief guest in the morning session. Now the valedictory session has been concluded. Now I request Ms. Aditi from Psychology Department to give the prize winners list.
Good evening, everyone. I welcome you all to the much awaited prize distribution ceremony of Samagatha 24-25. Before we begin, I'd like to inform all the award recipients that as I call out your names, please come on stage to receive your certificates. Once you have collected your certificate, kindly proceed to the back of the auditorium where you will be collecting your cash prizes. I request all of you to ensure a smooth ceremony. We start with interclass competitions of Samagatha held in 24, starting with the crossword event held by the Department of Mathematics. The first place goes to Vrishti J and Keetana R from 1st BSc PCM. I request Dr. Saujanya ma'am to please give away the prizes. From first BSc CBZ. And the third place goes to Nisha Sharma and V. Janhavi Murthy from third BSc. Farida, third BSc, and the third place goes to Varsha V. Gowda, second BSc. Science charades organized by Biotechnology and Zoology. First place goes to Priya S, Varshita, Bismi, Jasmine from second BSc. And the second place goes to Vatsala, Shailaja, Shri Tanaya and Pranati S from third BCA. Computer Science Department. The first place goes to Ashia Banu and Afia Banu from 3rd BSc. And the second place goes to Chetana VB and Kavyashri B from 3rd BSc. The first place goes to Sanjana Singh and Anushri from 2nd BSc. And the second place goes to Divya Rani and Muthu Lakshmi from 3rd BSc. Thank you, ma'am, for giving away the prizes. Now I ask Sharmishta ma'am to give away the prizes for our Intercollegiate Fest. Crossword was organized by the Mathematics Department, and the first place goes to Tarindu Bandara from Reva University. And the second place goes to Nrupatunga GH, again from Reva University. And speak by Botany Department. The first place, the winner is Radhika J. Jadhav from University College for Women. The next event, Science Charades by Biotechnology and the next event, Treasure Hunt by Psychology Department. Give them a big round of applause. By the Physics and Computer Science departments. The first place goes to Radhika from University College for Women. And the second place goes to Harshita N and Nazmeen Kausar from University College for Women as well. Please give them a big round of applause. Connections organized by Chemistry and Biochemistry. The first place, the winners are Harshita D and Vedavati S from University College for Women. We now have the prize winners for poster presentation. The first place goes to K. Jayashree and Dr. Sumalata from BMS College for Women. Then we have Devika S. Shravani K. Prasad.
Good evening all. I know this is not the time because it's already late and you people want to get back home. So let me make it a very short one. So respected our speaker for the session evening, I think we had Dr. Saujanya Madam, and all my dear faculty, especially the faculty from MLAC, those who have participated here. We also have some important judges who have judged the poster presentation over here. So we have Dr. Chitra over here and another, Prajanya, Miss Prajanya, she is also here. And they have taken the, patiently they have gone through the posters and they have, that is how they have evaluated. As we gathered here for the concluding session, that is we are into the valedictory, I should appreciate the MLAC faculty for the kind of effort they have put in. So let's give a round of applause to all the faculty. I would not say only science. All the 21 departments have joined together because the topic itself was so interesting that it had to be all the departments with all subjects to be contributing towards artificial intelligence and the future. That is what we were looking into. So the topic as artificial intelligence, technologies and innovation for sustainable future was the apt topic for the present scenario of the society where we are looking into, and how we could sustain our environment, how we could sustain our the present situation. We have to work towards that because it is not just computer related, but it should be related with various other aspects, how we could incorporate AI into various fields. That is the important task. I think our one day the conference was totally subjected to. So today we have really witnessed the rich ideas and perspectives and experiences from the keynote address to various speakers. We heard from them and we got to know that how we could work, including having AI into our field and how it can be taken forward. So this is something which we have to look into because this is the way ahead and we have to look forward for that. So latest advancements of AI, how examining its applications and implications of it and its sustainable development is the most important aspects as we look into. So I would like to thank each and every one of you, those who have participated from other colleges. Many of the participants have come, and those who have engaged, organized each and every event, yesterday's events, the departments, those who have were participating in various committees. It is not just 1 or 2 days, but it was months together you people have put in together. Where is, the students, volunteers who have came together, who have worked towards this. So I would like to thank each and every one of you for contributing for today's, conference. So to our participants, I hope this knowledge, ideas and connections have gained from this conference will take you to explore more about this aspect, whatever we have worked today. So with all these words, let me congratulate MLAC faculty for being a part of this Samagatha 2025, and let us continue to work together to harness the potential of AI and for the sustainable future. with all these words, let me congratulate once again all the faculty, and I should thank our organizing science coordinator and all the organizing team, those who have sat together days after days to make this conference a success. So let me thank all of you and all the best for your future endeavors. Thank you.
Token of appreciation to Dr. Saujanya ma'am. Prajanya ma'am to come on dais to give appreciation to our judges. Dr. Chitra Ravi ma'am.
पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Ready?